Good afternoon, ladies and gentlemen, and welcome to Clinical Trials Day of SALHN Research Week. My name's Simon Windsor. I'm the Manager of Research, Governance and Ethics here at SALHN. And today's first session is titled, Why Do Clinical Trials at SALHN? Thank you for joining us here in person and online. We acknowledge the Kaurna people, the traditional custodians of the Adelaide Plains, and pay respect to elders past, present and future. We recognise and respect their cultural heritage, beliefs and relationships with the land, and we acknowledge that they are of continuing importance to Kaurna people living today. So we do have the Hoover event platform, and you've got the ability there to ask questions. We've also got the ability to ask if people have questions here in person. We can do that as well. But yeah, it's great to interact on the Hoover app there. And we've also got the active research trail, so we've got the QR code, and that does enables your phone to interact with the 4 fields of inquiry logos that we have. And then following this, we'll have a poster session up there in the foyer. So that the posters are also available on the Hoover app, so you can have a look at today's posters and the rest of the week's posters at your leisure as well. And just wanted to highlight, we have a really engaging program this week. This afternoon we've got the Aboriginal Torres Strait Islander Research panel, ways of knowing, ways of being, ways of doing. And then tomorrow afternoon we have the Chalmers oration as well. So I welcome everyone to go to that as well. So now we can move on to the first session, and our facilitator for the session is Jenny Richter AM. She's a member of the SALHN Governing Board and an absolute research advocate in South Australia and Australia. So can we have a round of applause for Jenny?
Thank you, Simon. And welcome to day 3 of the SALHN Research Week. And what a fabulous event we've experienced so far. Clinical research covers a range of activities in which people or data or samples of tissues from people are studied to understand health and disease. Clinical research helps find new and better ways to detect, diagnose, treat and prevent disease. Clinical research can include disease mechanisms, translational research, clinical knowledge, detection, diagnosis and the natural history of disease, therapeutic interventions including development and clinical trials of drugs, devices and instruments, primary and secondary prevention and health promotion, behavioural research, health services research including outcomes and cost effectiveness, epidemiology and community-based and managed care-based trials. So clinical research covers just about everything. Clinical trials, on the other hand, are an important part of clinical research and will always involve an intervention or combination of interventions. These interventions may include drugs, devices, diet, lifestyle, or other behavioural modification programs. Clinical trials study new tests and treatments and evaluates their effects on human health outcomes. People volunteer to take part in clinical trials to test medical interventions, including drugs, cells and other biological products, surgical procedures, radiological procedures, devices, behavioural treatments and preventative care. So clinical trials, a very important aspect to our clinical research. At SALHN, our clinical trials activity is good, but it could be better, and we would like certainly to see it grow even more. In this session, we will hear from Professor Chris Karapetis, a practising medical oncologist. Professor Karapetis is Network Director for Cancer Services here at SALHN and is also Head of the Department of Medical Oncology and Director of Clinical Research at the Flinders Medical Centre and Flinders Centre for Innovation in Cancer. Professor Karapetis has been the principal investigator of over 160 clinical trials. It's a very impressive history. His interest in clinical research developed following a research fellowship at Guy's Hospital in London. He has established research interests in the areas of lung cancer, gastrointestinal malignancy, molecular targeted therapies, predictive biomarkers, epidemiology and clinical research methodology. Professor Karapetis will present to us for about 30 minutes on why do clinical trials at Southern, following which we'll have a Q&A session. Please log your questions through the Hoover app, and we will also take questions from the audience as well. Please welcome Professor Karapetis.
Okay, great. Thank you, Jenny. So I thought what I'd do in the next half hour is give you a presentation in sort of two parts. The first part will be really around answering the question, why do clinical trials in cancer full stop? And I'll give you a sort of a brief history of research in cancer from antiquity until more recent times. And then for the second half, I'll talk about why do clinical trials here in SALHN, and just talk about the sorts of work that we're doing here through the Flinders Medical Centre mainly. So, yeah, so I've titled it The Path Forward. So I'm going to go back to 400 years BC. This is Hippocrates, the father of Western medicine, who thought that diseases were really caused by an imbalance of four bodily fluids, the humours. He talked about blood, phlegm, yellow bile and black bile. And when he talked about the malignant state, he blamed it on a build-up of black bile. This was a theory that stood for 1900 years. There was little in the way of inquiry between 400 BC and all the way into the Renaissance period. When scientists in the Renaissance period, the people that were interested in inquiry and enlightenment, started to do things like anatomic dissection, and they went looking for the black bile that they thought was the cause of illness, and they couldn't find it. So that theory started to be questioned. In the 1700s, we thought that cancer was caused by a build-up of lymph, coagulated lymph. I think that was based on the observation that many cancers will cause lymph gland enlargement. In the 1800s, though, we really started to move into the modern era of cancer research. And the concept that cancer is a disease of cells came to be understood. There were many researchers that were exploring this. One of the most well-known is Rudolf Virchow, and these are some of his pictures. So we started to see the use of technology, microscopes, and an understanding that cancers are made up of lots of abnormal dividing cells. That's 1800s. I think a lot happened really at the end of the 19th century and the beginning of the 20th century when it comes to cancer research. In Europe, they started to develop models of propagating tumours in mice. And that became a really good model to actually test various cancer treatments. Tumour transplantation from one animal to the other was reported. We started to see the use of radium and X-rays. It was in its infancy, but it started. In the early 1900s, the concept of carcinogenesis being a multi-stage, multi-factor process came to be. The discovery of viruses in chicken that could lead to DNA damage, that could activate oncogenes, that could cause cancer was being reported. And Theodore Baveri, who was doing work looking at chromosomes in Drosophila in insects, came up with the theory that chromosomes carried important information and that cancer was due to a defect in those chromosomes. So these were all theories that were coming to be about 100 years ago. Private endowments were also starting to fund research, particularly in the USA and in Europe. And so research started to be supported, and governments were starting to look at research as well as they saw a value in it. So the last 100 years, as I say here, really overshadow all of the years that come before. And we can say that about a lot of things when it comes to technological advancements, and I can certainly say that about cancer and cancer research. A lot has happened over the last 100 years, sort of since then. In the late 1800s and early 1900s, the research was sort of mainly around cytology, and in the early 1900s, an understanding of genetics. Between 1920 and 1950, it was more about biochemistry and kind of chemical discovery and discovering drugs that have an impact on cancer cell growth, and I'll talk about a couple of those. From 1950 kind of onwards, a lot has been discovered about DNA and nucleic acids. And 1980 onwards, receptors, things that sit on the surface of cells that are involved in regulating the way a cell behaves and grows, and the signals that they send through the cell, so-called signal transduction pathways, came to be known. And studies looking at gene transcription and epigenetic control of gene expression also started happening, with a better understanding of whole system biology, how these things all work together. And more recently, we've seen our understanding of the immune system and how it interacts with cancers and cancer cell growth turn into treatments that are making a big difference in the treatment of certain types of cancer. Now, my focus is really on medical therapies for cancer, and I wanted to bring into the discussion the beginning of medical therapy for cancer. And I'll talk a little bit about nitrogen mustard. Nitrogen mustard, as you know, was used in World War I as a weapon. But there were observations that people exposed to nitrogen mustard developed low blood counts and a reduction in lymph nodes. In World War II, it wasn't used, but it was being researched, particularly in the US, as a potential treatment for certain conditions that caused enlarged lymph nodes and high blood counts. There was actually an accident as well that I read about in World War II off the port of Bari in Italy, where American soldiers were exposed to nitrogen mustard accidentally, and they again got the low blood counts and a depletion of lymph. So these studies were actually being conducted through World War II, and in fact one of the major studies was completed in 1943 that showed that lymphoma can be treated successfully, or you can get a good response, I should say, with nitrogen mustard. The results weren't published until 1946 because it was all done under a code of secrecy through the war. And what started happening then is that nitrogen mustard started to be used by certain clinicians as a treatment for lymphoma and leukemia, but the responses were short-lived, and it was a difficult environment, the 1950s, to be using these sorts of treatments. It was sort of seen with the negative light of the war experience. It was seen as being very, very toxic. There were other drugs that were developed around that time that had a similar effect, certain antibiotics and other medications that had an anti-tumour effect, but they were all very toxic. I'll mention methotrexate here, which was first licensed in 1947. That was the first medical therapy that was demonstrated to cure cancer. It cured choriocarcinoma and gestational trophoblastic disease. So that was in the late 40s. And again, that comes through that period of chemical discovery and the importance of folate metabolism on cell growth. So that was 1947. As we go into the 1950s, I mean, there was clearly a known sort of poor outcome with advanced cancers. Treatments were being developed, and you were getting these short-lived responses, not particularly long-term responses. We needed to work out how to best use these potential effective drugs, what doses, how much, how often, and what sort of combinations. And there was acknowledgement that these drugs had major risks as well. And as I said before, the environment was hostile. The doctors that were trying these sorts of treatments were often ostracised and considered to be sort of quacks and crazy for putting people through potentially harmful therapies. We needed clinical trials and we needed evidence in cancer. So, we'll talk now about what a clinical trial actually is. This is there are various definitions, but this is a definition that comes from patients living with cancer. That's the website. It's a research study involving people that tests new treatment and prevention methods to find out if they are safe, effective, and better than the current standard of care or the best known treatment. And we all recognise the importance of a randomised controlled trial, because when we're looking at a new treatment, we want to kind of demonstrate that it really is better than what has come before. And so we need to have some sort of a comparator. And we all understand the problem of confounding factors and biases. Some of the very early trials were comparative but not randomised. The very well-known study of oranges and lemons to treat scurvy was a comparative study in 12 people, I think, which showed that oranges and lemons worked. And there were other studies that looked at vaccines and smallpox and medical therapies for cholera that were comparative but non-randomised. The first placebo-controlled randomised controlled trial was looking at antibiotics to treat TB. and that was in 1948. So childhood leukemia, up until the early 60s, was a diagnosis that had a terrible prognosis, very short survival, almost 100% mortality. There's an interesting experience that you could read about of Nason's anablast, which just talks to the quackery that was sort of going on when it comes to looking at diseases that have poor outcomes, you know, obviously with childhood leukemia, a lot of emotion. Nason was a biologist from France who developed a serum from horses that he said contained a chemical that would wipe out the leukemia cells. And he gained a lot of attention. And there was the case of Edward Blake, a boy from Blackpool, who was flown over to France to receive this treatment. And what followed was a lot of media attention. There was a microscope sort of shone on the fact that the British system couldn't really develop leukemia therapies. There was this sort of negativity around the whole diagnosis. There was little media attention really on things like this because it was just so negative. And what transpired was an analysis, a proper analysis actually done by the Institut Gustave Roussy in Paris of the anablast treatment approach, which showed that it was just quackery. It just didn't do anything. It had no biological effect. But it just emphasised the point that proper research was needed, because when you're in that kind of environment, it does sort of encourage that kind of extreme, as I say, sort of quackery and charlatan type practice. We needed research, we needed evidence before we could actually recommend a treatment that does have significant risks, side effects of chemotherapy. And in the US, the National Cancer Institute Clinical Center at Bethesda in Maryland announced in 1964 that they developed a multi-drug regime for acute leukemia that seemed capable of killing every leukemic cell in a small proportion of human patients. That was the start of curing leukemia, 1964. But it was a difficult beginning because, again, these treatments caused hair loss, made children feel sick. Some children got very sick. Some children even died from complications related to the treatment. And so it didn't just automatically get embraced, and there were centres that took it on and tried treatment. There were other centres that actually focused more on supportive care. And this was, again, really kind of driven more by the families of the children who wanted the best outcomes. And what they did is they spoke with their feet and they went away from the centres that weren't doing the research, because these treatments were being done through properly conducted prospective coordinated studies. And they went to the centres, and these have ended up being the big childhood cancer centres, particularly in areas like the US and in Northern Europe, to get the best possible outcomes. And this was all done within a kind of a clinical trial framework. So there was a push for trials to try and get the best results. And we now know that with childhood leukemia, we get cure rates that are in excess of 90%. And that compared to sort of almost 0% in 1960. So this just talks to the more recent reduction we've seen in mortality. And this is looking at a younger group, patients aged 40 or less, over a sort of a 10-year period. And you can see that most of the reduction in mortality is occurring in the youngest age demographics. It really dips down to that young adult group. It lifts a little bit when you get into your sort of 30s. And what's interesting is it really matches very well with clinical trial accrual and participation. So those groups that have the best outcomes and the improvements in mortality are the groups that are getting involved more in trials. And the correlation is strong, it's very strong. Reduction in mortality correlates with clinical trial accrual. And this is looking at the same sort of statistics in lymphoma. Again, you know, in the younger groups, greater clinical trial participation, better outcomes. And it's really interesting to see that when you look at, you know, the less than 20-year-old lymphoma cases, the proportion of patients that are participating in clinical trials is sort of approaching 50%. Once you start getting into the adult group, unfortunately, the proportion is much lower. It's generally less than 10%. And this really talks to that as well, that we've got a significant degree of clinical trial participation in the younger demographic where we've had the greatest benefit in terms of mortality reduction, better long-term outcomes than we are achieving in those that are older. So it'd be great if we could lift clinical trial participation across the board, I think. So that takes us to Flinders. Now, when Flinders started, it didn't have medical oncology in the 1970s. It wasn't really a subspecialty that was accepted. It was just beginning. It kind of grew out of haematology and physicians interested in oncology and cancer biology and cancer treatments, but it wasn't a set specialty. And in fact, the first medical oncologist started here in 1990, and he really arrived at the request of the surgeons who wanted an oncologist to help treat patients around their breast cancer surgery, because then it was noted that giving chemotherapy after breast cancer surgery improved survival. So they needed somebody to give that chemotherapy and manage patients. So that was really the driver. We didn't have clinical trials at Flinders until the year 2000, which is when I came to work at Flinders 21 years ago. I had to put this up. This is a photograph from the year 2000 of what was the medical oncology department. Alison Richards on the left is still with us. Bogda Kotswara, many of you know, is still with us, and that's me, looking a bit younger. Margaret was our chief nurse. She's now retired, and Trevor Molden was that first oncologist who has also since retired. So we're kind of a stable lot. We're hanging around. So it started, clinical trials in oncology started at Flinders in the year 2000. I came along from Guy's, where I did a clinical research fellowship, and I kind of really wanted to help establish the clinical trials unit and run clinical trials here at Flinders. Now, there was a study that had just started, and I'll just talk about an interesting experience. It was the Amgen study looking at a drug called pegfilgrastim, which is a long-acting granulocyte colony stimulating factor, which has now become a standard. So this study kind of helped to provide the evidence that showed that this drug really worked, and it made it a bit easier for patients after chemo. Anyway, I got a patient onto the study, and at that time, I consented the patient myself. I quickly realised that we had no infrastructure. We had no research nurse. We had no one else to assist me. And that particular patient had a significant adverse event, a reaction in their lungs called pneumonitis. And I had to do all the legwork of reporting that adverse event and sending it through. It was all by fax then, it wasn't electronic. And it was a whole lot of work. And I quickly realised that we needed to really establish a unit, and that didn't take long. In June of that year, we employed our clinical trials coordinator, Alison Richards, who's still with us. She was partly funded through a Cancer Council grant. The unit quickly grew. We had 4 trial staff by 2005. 2009, we employed a medical research fellow, a research nurse, a dedicated nurse in 2012. In 2009, we were doing sort of 26 open studies. We've now got over 60 open studies. We've got 9 research staff, and we're expanding to 12. This is our cancer centre, but I can tell you when we first started, we were just occupying rooms at the back of corridors and wherever we could find space. So we've definitely progressed into a much nicer surrounding, which is great. And these are the team in action on the left. This is the team in the cancer clinic. On the right there, you can see the team kind of working busily and at their desks. And at the bottom there, where start of the year actually, we got together for a bit of fun time, which is always, I think, really important when you're working in this sort of area. When we talk about cancer drug development, there are many phases from sort of, early scientific discovery, preclinical development, and then you've got the phases of the clinical trial, and we're involved in all phases, phase 1, 2, 3, and then you've got a period of registration type research to sometimes prove safety in a larger number of patients, and we get involved in those studies as well. So we're mainly involved in the middle of that graph. I wanted to sort of talk about a few of our wins and a few of the studies that we were involved with. This is looking at imatinib, a molecular targeted therapy that really kind of changed the game in cancer, where we could treat a cancer successfully by targeting a very specific molecule, a very specific pathway, and we could identify that pathway kind of up front. And we were seeing responses like the pictures show you there, a big mass in the pelvis disappearing to near nothing. And we were involved in some of the very early trials looking at treatments like Gleevec. I was fortunate enough to be involved in a big trial that I helped to coordinate as the Australian lead that looked at using a type of antibody in the treatment of colorectal cancer. We initially found that the antibody did provide a benefit. It helped patients to live longer. But we then found that that benefit was restricted to patients that had cancers that did not have a particular mutation in the DNA of the cancer cell. The KRAS mutation needed to be absent. And that was research that we led from here that led to a publication in the New England Journal of Medicine. And I like to think that that's kind of inspired other researchers around me. I'm fortunate enough to be working with the Precision Medicine Unit with people like Michael Sorich and Andrew Rowland and Ross McKinnon. And you know, this sort of success kind of leads to more success and more research, I think, that can come out of Flinders. I mention this as well because this is a study that we were part of. This was the pivotal study that looked at using an immune therapy approach to treat patients with melanoma. It was a difficult study because we were discovering immune toxicities. And particularly with this drug, a drug called ipilimumab, it can cause very severe diarrhea. And this was all happening through the study. We were getting kind of warnings, and we were very concerned about patients getting sick. But anyway, at the end of it, this drug was a drug that was a huge game changer. It improved the survival for a proportion of patients. And you look at that survival curve, and it does drop off. It's only 20%, but it stays flat. So we hardly ever saw survival curves that stayed flat. So a disease like metastatic melanoma had a kind of almost a 100% death rate at 5 years. We now had a 1 in 5 chance of surviving 5 years or more. And that's improved since then with the combination of more immune therapy drugs, where we're getting 5-year survival rates of 50% or more, when it was previously 0, really. And even phase 1 studies. Phase 1 studies were studies, and they still are studies, that really try to look at what is the right dose of a drug and what are the side effects and what is the tolerance level. And that's still very important. But we're starting to see studies like this, a phase 1 study where the bar going down means the tumour's shrinking. The bar going up means it's growing. And you can see here that the majority of patients in this phase 1 study had tumour that shrunk. I mean, that's quite exceptional for a phase 1 study. And the other thing we're seeing is studies like this. Again, bar going down means the cancer's shrinking. This is looking at lung cancer, treating it with a tablet, but looking at specific biomarkers. In this case, it's the ALK mutation. Where that's positive, you get quite an impressive response. So we're getting better at selecting treatments and giving more specific personalised medicine for that specific type of cancer and getting better outcomes for it. So it is complicated. It's very complicated. Cancer is complex, we know that, and we're only beginning to scratch the surface. But as complex as it is, we are getting some winning runs, whether it's through boosting the immune system or targeting one of these signal transduction pathways when it's found to be the important pathway or the driving pathway for that cancer, and we're getting better outcomes. So this sort of complex biology shouldn't scare us. So the path forward, I mean, I can tell you when it comes to medical oncology, when I started in medical oncology and I went to my mentors and asked about sort of guidance, I was told that cancer was too hard, medical oncology was too hard, the disease was too complex, the specialty would be too emotional, soul-destroying. You're a smart kid, you could do better than this. And there was a real therapeutic nihilism. But it's changed now. Now medical oncology is becoming an attractive subspecialty. We've got some of the best and the brightest wanting to work in the area. We've gone from too hard, too complex to really a sense of scientific discovery translating into clinical improvement, improved outcomes, and a brighter future. Thank you.
Yes. Well, that's amazing. Thank you. It's great to actually understand the history and the evolution to put today's discovery in context, really, isn't it? And all about getting the evidence to ensure patients get good outcomes. I did a few maths while I was sitting there too. If you've been here 21 years,
Yes.
and about 160 trials, it's roughly about 8 to 10 a year.
Yeah.
Probably a bit more now. Yeah. So I think that's a fabulous contribution to the whole research culture of Southern. Now I have the prerogative to ask the first question. Sure. So my question is that, given your experience with clinical trials specifically, and the value that this brings to our clinical research culture and patient outcomes, what else could be done in Southern to grow clinical trials, and how can the board help?
Yeah, I think that's a good question. Yeah, no, it's obviously a good question, an important question. I think obviously we work within an environment, that's regulated and appropriately governed, and I think that's very important for research. And I know a lot of effort has gone into the whole governance sort of process around trying to make research happen, so trying to be proactive rather than being obstructive. I think there's a recognition that that this research is really all about improving patient outcomes, recognising that there are risks, and there are lots of factors that you need to take into account. But if we approach it, with that philosophy and mentality, I think we're going to find ways of overcoming sort of roadblocks and barriers. And I've certainly seen a huge improvement in the last 2 years, really, year and a half, around the process of putting through our applications and getting approvals, the reduction in the duplication of effort. The amount of... I used to sit on the ethics committee for 11 years, and I'd review a project that they'd review down the road, and they'd review at 20 other hospitals, and it'd be the same project, and we'd come through and we'd tease out little tiny things, and we'd kind of... I think we moved away from that now with the kind of multi-ethics approval systems that we have. So that was a big step in the right direction. I think that we need to be doing things that promote research. So that's kind of facilitating the research that's happening. But the promotion of research, I think, is still lacking. I don't think we recognise the research as well as we should do. And I don't think we really employ for research. So I'd like to see things like sort of benchmarks and regular reporting back to the executive about our research output. And that doesn't happen here. It happens in some states. New South Wales has the Cancer Institute of New South Wales, which is really well resourced, and they actually provide money to support research at various sites. But in return, you've got to report back annually. How many trials have you done? How many patients have you got onto the trials? What trials do they get onto? We don't have that level of oversight and reporting, and I think that would be a really good thing. And at the same time, within each department, within each unit, this is not just oncology, obviously, I think there needs to be the encouragement of researchers, of identifying young researchers coming through and allowing them the opportunity to work. We can look at high degrees, PhDs. I find that still isn't happening as well as it could be happening here at Flinders. So I think the systems are pretty good now, but I think the promotion needs to happen. And I think there needs to be a better way of capturing all of our activity.
On Monday, we talked a little bit about clinical academics. Do you have a view about whether we should be growing them, or is there a different way of actually promoting clinical trials research within the clinicians?
I think the clinical academic pathway is a good way. I think where you have people that are employed, again, it comes back to making it part of the job. It's an expectation that you'll teach and that you'll research. At the moment, we're really kind of measured more by our activity around clinical service if you're working with SALHN. Certainly in medical oncology, that's how we feel, and we've got a very busy clinical service. And that's important, but we're not being measured against our research output. No one's asked me for my publication record, for instance, or for the department's publication record. I think we should. No one's asking me about what our kind of junior doctors are doing as they progress through their sort of career and they advance professionally. I think we should. I think if you have the benefit of the clinical academic system is that you will kind of protect, I think, that research and teaching and that academic part of the job through that kind of an appointment. So I'd be in favour of that. But I think even without that, I think SALHN should be looking at ways of sort of supporting that kind of work.
Yes,
I think it's important. And you know, we can see through a whole host of examples that those sites that do the research are the sites that have the best outcomes. And that's really important.
Excellent, good advice. Now, to some questions. And I'm glad this question's been asked. Why are clinical trial participation rates higher in children than in adults?
Yeah, so I think it's because of what I said before. I think there's a recognition that clinical trials lead to the best results. I think when we're always trying to improve on things. It's getting a bit difficult now in children because they've reached that ceiling of over 90%. But when they were down at 0%, 10%, 20%, 5-year survival, I mean, that's poor. And so everybody said this should be better. And I say that too with patients that I'm seeing now who have a cancer with a poor 1 year, 2 year survival. I tell them the treatment that I've got to offer you, the standard treatment is simply not good enough. We need something better. Now, I think in the children's example, there was a clear recognition from families that that treatment that they had wasn't good enough. We needed something better. Now, when you are going to do something better, it's going to be something new. And when you do something new, you need to do it in a way that is organised, well-defined, it's structured, it's a protocol. I mean, even now, even if they don't have the protocol to treat patients at the kids, there isn't an active study, they still treat patients according to a protocol. It defines a clear way of managing, well-defined, and managing complications and everything. It's a rule book. So I think with kids, there was a clear push towards better outcomes, and the best way to achieve that was through new treatments, and the only way to really test new treatments is through clinical trials. With adults, I think there still is a degree of nihilism, especially around certain cancer types, advanced lung cancer, advanced pancreatic cancer, where our clinical trial participation rates are really still very, very low. So we need to find better treatments.
So is it the participation rates, the older patient doesn't want to participate, or do we not go out and actively pursue them?
I think it's changing. I think our older patients are getting remarkably fitter and cluey and wanting treatments, and so those kind of ceilings of age don't seem to apply much anymore. I'm seeing people that are 80, 85 are remarkably fit and cognitively well. We obviously take into account comorbidities, and that's all really important. And yeah, and as you get older, comorbidities come into play, and suitability for trials then goes down. So there will be a reduction. You don't have the comorbidity issue in children. So that is a factor. And look, as people get older, you know, there is, I think, an acceptance of mortality in the end. And so you will get people who are more likely to say no to certain interventions. So I think the participation rate will be lower, but I think it'll still be a whole lot better than 3% or 4%.
Yeah. Because there is a correlating question here about how do we identify or measure consumer demand for specific clinical trials?
I think you need to engage with consumers, and that we're seeing a lot more of that. We're certainly seeing it at the research level now, so when we're putting in our grant applications, we need consumers on board to look at our projects and to feedback as to whether or not what we're doing and what we're looking for matters to them. I think that's really important. I've just put in an application where the consumer on the application was a chief investigator, and she was amazing. She did so much work to help the application really develop. So yeah, we need to have that kind of level of engagement. And there are organised groups, Cancer Voices, SA, and other groups that you can turn to for that kind of input. But that's what we need. We need more of that.
There's a question here about how do you choose which clinical trials you want to conduct at SALHN?
Yeah, look, so that's a good question too. I mean, at the end of the day, you know, you look at the disease, you look at the trial that's on offer, you look at the treatment options, and sometimes we'll see a treatment or a trial that doesn't seemingly kind of add to what we're already doing or where the aims look to be not particularly, encouraging, and we may say no. The standard of care may not match our current standard of care. So if you're doing a randomised trial, we may be offering people a treatment that we believe to be inferior, so we'd obviously say no. The trials that we do, trials that could be, we could be invited to participate in through a pharmaceutical company developing a drug. So we need to look at the drug and what kind of evidence they have and where this sort of treatment's going. That was certainly true of the immune therapy treatments, so we're pretty excited about those when they came through. But many of the trials that we do through cooperative study groups. And those cooperative study groups have kind of working parties, big groups that look through various trials and actually select out those that they think are the way forward. So there is that kind of expert oversight, and we often get involved in those. And again, through Flinders, we have people, myself included, that sit on those working parties with the various cooperative study groups. So it is a complex process, but I'd have to say for some cancers, I just wish we had more trials coming our way, because for some areas we just have very few trials. And the big area are kind of the rare tumours area, where unfortunately the commercial reality is that pharmaceutical companies will feel they won't get a benefit, and we don't have the degree of coordination that we do, for instance, when it comes to breast cancer or colorectal cancer. We don't have those cooperative groups. So there is a group there that kind of misses out, I think, the rare tumours.
Because the almighty dollar is always there in the background, isn't it? And so can you talk a bit about the relationship between the pharmaceutical companies and the clinical trials and how we would interface?
Yeah, no, so that's right. So these trials, if they're successful, there's a huge commercial gain at the end for the companies. And so they invest in the trials, and I hear various figures from sort of 300 to 900 million, the cost of developing a drug all the way through. If it makes it all the way through, many don't sort of get all the way through. So if a trial is sponsored by a pharmaceutical company, they will support the conduct of the study. And effectively, our clinical research unit is funded entirely through a special purpose fund that is built on that kind of funding through pharmaceutical companies supporting studies, through even the cooperative group studies. They would receive some funding, often coming through pharmaceutical companies, that would filter through to the centre here to conduct the studies. And we're lucky enough to have also been successful in grants, and these are sort of government grants and other grants that come through to support our studies. So effectively, we've got a unit that we've developed where we don't need to tap into any operational funding, and all of that sort of keeps the research going. But as the research increases, and that's what we've seen, we've needed to grow our unit. Some of the trials are getting much more complicated, where we start looking at biomarkers and specific subtypes, and we have to screen sometimes 100 patients to find the one that has a specific biomarker that could go into the trials. There's a lot of work that goes into selecting out one patient that eventually goes in, but the outcome for that one patient can be great. So as long as we're resourced to kind of do that, we'll do that.
And is it the volume of patients participating in the trial is obviously a challenge at some points.
Yeah.
Now, there's a really interesting question here about any plans or thoughts for complementary and... Oh, beg your pardon, sorry. There's a really interesting question here about whether there are any plans or thoughts for complementary and alternative therapy focused clinical trials.
Yeah. So we're certainly interested in examining and exploring all aspects of therapy when it comes to treating patients. If it's a complementary approach, again, we want that approach to be evidence-based. So I'm just trying to think through the sorts of studies that we've done that have kind of fallen into the complementary therapy space. It depends. We've been looking at trials that explore the benefits of cannabis and cannabis-related products. Whether that's complementary therapy or not, I call that drug therapy. But that probably won't come into that. But yeah, I'd be very happy to look at studies that explore that. Those studies won't come with pharmaceutical company backing, but hopefully they'll come with grant funding and some sort of support in order for us to be able to do them. And we sometimes run studies that we could kind of fund within our existing resources. We've done studies that look at, for instance, long-term effects of treatments that we've had no funding for, but we've managed to sort of make them work within the resources that we have at hand. But complementary therapy is certainly an area that we could get more involved in.
Are you seeing patients actually asking for complementary therapy alongside the more traditional therapy?
Yeah, I mean, that's always been the case. So again, we kind of adopt an evidence-based approach. But, you know, most would be permissive of other treatments as long as they're not going to interfere with the existing treatment. So we usually want to see what patients are receiving, and most of the time the complementary therapies, if we're talking about, you know, vitamins and things like that, they're not going to be harmful. But there are other complementary therapies, yoga, massage, exercise. So we did do an interesting study of exercise, now that it's come to mind, where we looked at the use of exercise as a measure, as an intervention to reduce the risk of bowel cancer recurrence after bowel cancer surgery. We worked with our physiotherapy department. We were one of many centres. It's a big, large, clinical trial, a randomised trial, again, where half the patients just got told exercise is good for you. The other half got told you need an exercise prescription. You'll see a physiotherapist, they'll work out your baseline fitness level and they'll prescribe an exercise program. That study closed about 3 years ago, and so there's a large group of patients that have participated from Flinders who were part of the follow-up of that. And we're trying to prove that the group that got exercise is going to have a lower chance of cancer recurrence. And you need that kind of evidence to kind of support the resource, because if we do find that exercise makes a difference, it saves lives, it may save lives to the same sort of magnitude of benefit as chemotherapy does after surgery. Well, then we should resource physiotherapy. We need to kind of, that would be used as an argument to resource that kind of intervention. So you can sort of see that as complementary, but it's an important study, I think.
So are there any questions from the audience here? No? Okay, I've got one. Yes, thank you.
Thanks, Chris. Great talk. I had a question regarding, you sort of touched on it before, that modern oncology trials can be incredibly complex. You can have combinations of therapies, you can be looking for specific mutations in patients. How do you ensure that participants understand the trial that they're getting into? What goes into that sort of verbal explanation to them in that process of ensuring that they understand exactly what's going to happen and why it's going to happen?
Yeah, that's very important. So I think the usual starting position is you sort of, if you see somebody, you obviously get to know them, get a sense of what's going on and work out what you think the best course of action is, and you'll have a recommended treatment plan. And it might be, I recommend that you have a course of chemotherapy after your breast cancer surgery to reduce the risk of cancer recurrence. So you've got a starting point that relates to the standard of care effectively, and you've got to really explain that very well. Now, if a patient has decided they want to go ahead with that standard of care, so you're in that space, that's when the clinical trial becomes an option. And so you need to then explain what is involved in being part of a clinical trial. You need to explain why the clinical trial is being done, but you also need to explain very clearly what the patient is going to do, especially the things that they're going to need to do that are in excess of what they would otherwise be doing. And you need to explain what the potential risks are. Now, we do that verbally. We do that, in our clinic. We have research nurses that do that as well. We encourage the patients to take a pretty detailed information sheet home to read through with them, their family, the people closest to them, their GP. And the decision's never made in a kind of a fast or rushed way. We don't normally need to rush these decisions. They aren't like sort of acute myocardial infarct studies. They've got time to think through things. Patients have the opportunity to ask questions about the trial. So this is really the process of informed consent. It's critical. And only after you've obtained full informed consent, patients can participate. They should know that they can withdraw at any time if they don't want to continue with the trial. But that's the basic process.
Any other questions?
I've got the microphone, so I might as well ask my question. What advice could you give to other departments at SALHN that want to establish some sort of research infrastructure? So what sort of steps would they take?
Yeah, so sort of from my experience, yeah, you do need a kind of an infrastructure to help the research kind of happen, but sometimes you've got to make a start and then you'll kind of watch it grow. So small steps, but steps nevertheless in the right direction. So I think when it comes to research, you need to think through what you want to be doing research in. You need to look through the feasibility of that kind of research, and you need to kind of surround yourself with people that would come along with you. Because we don't do clinical research as a single kind of mad scientist researcher. It really is a team, a group effort. So if you can build that network, that collaboration, start the research, work out exactly what's needed. It may be a research nurse, it may be a data manager. You need to look through what kind of resources you need, whether it's additional funding to employ more people, whether it's equipment. You know, these are the sorts of things you need to think through. But I think that if you make a start, and I'd be encouraging everyone to make a start, I think everybody has something that they're interested in, it will kind of grow.
Good. Chris? I've been given the nod that I've no more questions. Time's up. So, Chris, thank you very much. That was an excellent presentation. Would you join with me in thanking Chris for his time? That concludes this afternoon's session, but we're now all heading out to look at the posters. Correct?
We're going to free paper session next.
Okay. Did you want to say anything about that? No?
Good afternoon, ladies and gentlemen. Welcome to a really exciting session. My name's SALHN Windsor. I'm the Manager of Research, Governance and Ethics here at SALHN. We just had an excellent presentation from Chris Karapetis, the medical oncologist here. And so, yeah, this is the free paper session. We get to see what we're doing here at SALHN, which is really exciting. We acknowledge the Kaurna people are the traditional custodians of the Adelaide Plains and pay respects to elders past, present and future. We recognise and respect their cultural heritage, beliefs and relationship with the land. We acknowledge that they are of continuing importance to the Kaurna people living today. So we have the Hoover event platform, and you can ask the presenters on this platform questions from their presentation. So if anything comes to mind, we do have a short portion of time at the end of their presentation for you to ask questions. And we've also got the active research trail, and the idea that there is to basically describe some of the elements of our 4 fields of inquiry for you. After this session, we have posters which will be presented out in the foyer there with some lunch. So welcome everyone to join us for that. And then finally, we've got some prizes as part of this. So part of through the generosity of the Flinders Foundation, they've awarded, donated some prizes for the week. So today you can vote on the Hoover app. It's just a matter of going to today's session, and we've got a poll, and very sort of democratic, above the line style. You got one vote, and you can select which free paper presentation should win the prize. So that's each day of Research Week for each of our 4 fields. And just to reiterate, this afternoon we've got Aboriginal and Torres Strait Islander presentation. Tomorrow we're at Tonsley at the Innovation Precinct there, and we've got a really interesting session on data. The Chief Medical Information Officers there as well, with Israel and American partners that are working with us. So please join us for the rest of the week. Now I can move on to our facilitator for this session, Associate Professor Rosalie Greville, Consultant Obstetrician and Maternal Fetal Medical Subspecialist here at Flinders Medical Centre, and the Director of the Medical Program at Flinders University. So can I have a round of applause for Rosalie?
Thanks, Simon, for the introduction and for asking me to facilitate the session today. It looks like a really great session. So just for the audience's information, we'll have 3 minutes at the time at the end of each talk for questions, presuming the speakers don't run too over time. We'll try to make sure we don't run late. And we will ask you to, if you're in the audience in the room here today, to indicate you have a question. We've got some microphones so that the people who are online can hear the questions as well. So that's an opportunity to ask some clarification questions. So without any further ado, we'll introduce the first speaker, who's Professor Michael Shanahan, speaking on his topic, supracapsular nerve block treatment of adhesive capsulitis. Thank you.
Well, thank you for inviting me to talk about some of our research that goes on in our department. This is a study which has been recently completed that had been running for a number of years in order to adequately recruit, and we have finally got the results of the study, which we're very excited about. So this is a study entitled Suprascapular Nerve Block for the Treatment of Adhesive Capsulitis. Adhesive capsulitis, or frozen shoulder, a lot of people know it as frozen shoulder. It's a really interesting condition. It's not rare. It's about 4% of the population get this at some stage in their lives, slightly more women than men. A bit more common in certain populations, and in particular in people with diabetes. I see Morton in the room, and the endocrinologist would be quite familiar with this condition. It occurs also spontaneously and without any underlying predisposition. But it's a strange condition. It's an autoinflammatory condition. It has a fibrotic component to it. And what it is it's given as a description as characterised by a painful restriction of the glenohumeral joint, which is the main joint of the shoulder, in all planes of movement, both active and passive, in the absence of joint degeneration, which is sufficient to explain the restriction. It goes for months, so people develop a painful shoulder. It then becomes stiff, and then it eventually thaws. That's the pattern that we see. Unfortunately, quite a few people that develop frozen shoulder never completely thaw, and they're left with stiff shoulders forever. That's something that's not well understood. But even the average person with frozen shoulder, it will go for years if it's not treated. The prevalence, as I said, is around 4%, and it's around about the middle years where we mostly see it. How is it treated? Well, the bottom line is it's treated badly. So we treat it, we try and get pain relief, we try and improve arm movement and get people shorten the duration of disease and get them back to normal activities. That's the aims of the treatment. There are multiple options, and when in rheumatology, at least when you hear there's multiple options, it generally means they don't work. So there are multiple options for the treatment of frozen shoulder. Injection therapies, manual physical therapies, surgical therapies, radiological therapies. But they're all pretty unsatisfactory. There's been a recent review looking at all of this, and the review came out to say that the only thing that works better than placebo is an intra-articular steroid injection into the glenohumeral joint, if done early, and followed up with physiotherapy. It's the only thing that's different from placebo in terms of outcomes, as far as we know in the studies. And there's no consensus about the overall management of the condition and the sequence in which treatments might be offered. So it's a disappointingly treated condition. So the suprascapular nerve is a really interesting nerve for us because it's accessible in the clinic for treatment of shoulder pain, and we use it for a number of indications for shoulder pain. We've studied it quite a bit in our department. We have looked at the data available for frozen shoulder and the use of suprascapular nerve block. There's one or two little studies. They're short term. They're a single injection with a single follow-up. Encouraging, but certainly there's the evidence for the use of the nerve block in this situation is quite weak. How do we do the nerve block, just for your interest? Well, it's done with anatomical landmarks. We palpate the spine of the scapula, which is this structure here, and we draw a line. That line should be a little bit higher, and we palpate the angle of the scapula, and we draw a perpendicular from the ground up that way. And in that region is where we put the injection. You can see the view here. We put a needle down into that fossa and inject local anaesthetic and steroid, and it cuts pain at the shoulder for months. It's a really interesting treatment, and so that's how we do it. So our aim of our study was to study the efficacy of this intervention, the suprascapular nerve block, in the management of adhesive capsulitis. And we developed and designed a randomised, double-blind, placebo-controlled trial of standard therapy versus standard therapy plus the suprascapular nerve block. So in the standard group, people got the glenohumeral intra-articular injection and follow-up physiotherapy. And then every 12 weeks, we did a sham injection that mimicked the suprascapular nerve block, working from behind the patient so they couldn't see us. And the metrologist that followed the patient up was blind to the injection also. In the active group, they got the glenohumeral joint injection physiotherapy, and every 12 weeks until resolution, they got a suprascapular nerve block. So we included people, adults, who had a painful stiff shoulder for greater than 2 months with restriction in passive and active range of movement of greater than 30 degrees in two planes. That's the definition of frozen shoulder. We measured this with an inclinometer, our metrologist, and a rheumatologist reviewed all the patients to be happy with the diagnosis. We did an X-ray and an ultrasound at the start to exclude glenohumeral joint OA and also a bad rotator cuff injury. We also excluded people that had a lot of pain at rest or who had a systemic inflammatory condition such as rheumatoid, or the X-ray ruled out a fracture, obviously. If they were allergic to any components, they were pregnant or likely not to comply with follow-up or unable to understand the informed consent. Our main outcome measure was time to resolution of the frozen shoulder. How do we measure the resolution? Well, we used a series of measures. The first was the Shoulder Pain and Disability Index, which is an index specific for the shoulder that measures pain and disability, and a visual analogue scale, and range of movement. So we had several parameters of range of movement. And also, importantly, we included a measure of patient perceived recovery. We wanted to ask the patients, do you think you've recovered? Rather than just measure a range of movement. We wanted the patients to be happy with the recovery. So we defined resolution by saying if you got your abduction to greater than 160 degrees, so if you could get your shoulder up to here, if you could get external rotation greater than 45 degrees, this is the main movement we measure, or one of the main movements in frozen shoulder, that's resolution. If your SPARTY scores drop by 40 on a 100 millimetre scale, or your VAS drop by 20 millimetres on a 100 millimetre scale, and also if the patients felt that they'd recovered, 4 out of 5 at least on a Likert scale, we said they'd recovered. Recruiting was difficult, it was challenging. We assessed 110 people over the time, randomised 54. We needed 25 in each group according to our power studies to get a satisfactory separation of results. We had 27 in each group. We excluded quite a lot of people because they didn't meet the inclusion criteria. There's a lot of confusion in the community about what frozen shoulder is. People would come with painful shoulders that weren't frozen. And so we had to exclude those people. So we ended up randomising 54 over the time, which was appropriate for our power calculations. These are the demographics. They're roughly equal in terms of gender balance in the groups. The age is equal, dominant shoulder was equal, and the study shoulder was roughly equal. And in terms of our baseline characteristics, you can see they all had quite restricted range of movements. So just to give you an idea, most of them couldn't flex past here, about 90 degrees. They couldn't get beyond about 60 degrees. It's a pretty stiff shoulder, and they were all like this. They had high levels of pain and disability, and the duration of symptoms by the time they came to see us was equivalent in both groups. This is our main result. So people who received the placebo injection took around 11 months from the time we first saw them to resolution. People who had the active treatment took about 5.5 months to resolve. So a big difference between those two groups, around 6 months difference in time to resolution. This is represented diagrammatically here with a survival analysis curve, and you can see that the active group in comparison to the placebo group, settled far more quickly. The next few slides I'll show you, I'll just run through them quickly, are the data that make up that time to recovery group. So first of all, range of movement. This is flexion. The group that received the active injection, their flexion improved much more quickly. Same with abduction, which is the movement out from the side of the arm. Same with external rotation. All of these movements improved more quickly in the active group. Their shoulder pain and disability, measured by the Shoulder Pain and Disability Index, quickly improved in the active group compared with placebo, and that improvement persisted through each of the time periods, which are 3-monthly intervals. Same with the pain scores. Both groups dropped because we're doing a glenohumeral joint injection into both, so they both got improvement, but the group that got the nerve block improved more quickly and in a more sustained way. And importantly, people felt that they got better faster in the active group compared with the placebo group. And it's interesting, the people that got into the active group were mostly really happy with what happened because they've been putting up with a lot of pain and stiffness for quite a long time before they saw us. They got some treatment which immediately improved things, and they felt much better. So, a few limitations on the study. Very difficult to recruit. I've mentioned that to you. We might have missed a few frozen shoulders of people coming to us because when you first got your frozen shoulder, it's a painful phase. We might have excluded people that were going to develop frozen shoulder. We can't diagnose frozen shoulder if you've just got a painful shoulder. We did ask them to come back if their frozen shoulder, if they stiffened up. Nobody did, so we assumed that we didn't miss many at any rate. We had a small sample, but it was appropriately powered. The strength of the study, we followed these people through to resolution. And this is really unusual in studies of frozen shoulder. Usually they're one-off interventions. So I think this was a nice design from that point of view. It took a lot longer, of course, because we're following each for sort of 3 to 6 months at least. Patient-focused outcomes, very important in clinical studies. And also, the interventions are really easy to do. They're in the clinic, they're fast, they're cheap, and they're effective. So that's another really practical part of this study. So just in conclusion, this study, we think, establishes that the suprascapular nerve block is a very valuable adjunct therapy for the treatment of frozen shoulder, and it reduces the disease duration by 6 months compared with what is standard best practice today. In addition, we have achieved really good reductions in pain, improved function, and we had really happy patients. This is in contrast to many available therapies which have modest outcomes, and some of the interventions, to be honest, are brutal, and I wouldn't recommend them, and patients hate them. So, suprascapular nerve block, in our mind, can clearly be recommended as a safe and effective adjunct therapy for the management of frozen shoulder. We acknowledge that and thank the Arthritis Australia for a grant to help us undertake the study, and we thank very much the patients involved in the study, and also the people that referred patients to us, which were GPs and physiotherapists, and our orthopaedic colleagues were very helpful in referring patients to us. There are a few key references. Thank you.
Thanks for an excellent presentation. Are there any questions from the audience? I don't have any online at this point in time. I might just jump in with a question while everyone's thinking about it. I get the impression that the population that you're seeing was perhaps patients that warranted referral to a tertiary clinic. Do you think then that the results are applicable to the broader population out there who might, just see their GP or maybe even just rock up to the physio with a sore shoulder? And do you have any information on other treatments that the patients were seeking or receiving at the time, and were they taken into account?
Yeah. Okay. Thanks, Rosalie. So you're quite right. We don't see many of these patients in our clinic. So it's not like they were coming to us, we had to get it out there that we were doing this study. So most of the patients came to us either through the orthopaedic doctors that run a great sort of musculoskeletal clinic with a rapid triage, and they'd see a shoulder and they'd flick it to us, or from local physiotherapists, or from general practitioners that were seeing the patient. So we don't actually see many in the clinic ourselves, but that's where they were coming from. Many of them had treatments before they came to us with hydrodilatation or injections. Your comment that was interesting, you said, painful shoulder. This is the common mistake that people make in the community. We're not just dealing with a painful shoulder here. It's a frozen shoulder, which is it's a different kettle of fish to a painful shoulder. So that's probably why we had quite a lot of people come to us that we actually didn't include in the study because they didn't meet the criteria for a frozen shoulder. We think it does broadly reflect the community because that's where the patients came from. We had a few issues with getting it out there because for a while we weren't allowed to advertise the study. It had to be all by word of mouth. That was earlier on in the study. Later on we were allowed to advertise. So.
Great, thanks. Are there any other questions? Otherwise, thank you for your presentation, and we'll move on to the next speaker. So the next speaker we have is Professor Malcolm Battersby, I believe, presenting on behalf of his team about improving access to psychological therapies. Thank you.
Thanks, Rosalie. And I'd like to congratulate the SALHN board and the previous CEO and Andrew Burston, who has led this whole research week endeavour. And I think it's such an important step forward in getting better outcomes for our patients at the end of the day. I want to tell you about for something that it's my stage of the career has been one of the most exciting new adventures in working out what's going on for people when they're going through a suicidal experience. And it's particularly driven by the great therapy that we've been provided through Sue Boyd-Brearley, who's down the front here. And if we do get a chance to have any questions, there's some really important observations she's made in this process. So this is a great, interesting group of people who are co-investigators, and some of those people are consumers. I don't need to name them all at the moment because we haven't got time, but this project wouldn't have evolved the way it did if it wasn't for consumer input, and I'll touch on that. So this is the background. Before I start into what the issues around suicide, the acknowledgements I do want to make are about that we are meeting on the land of the Kaurna people, and we've already had that acknowledgement, but that's really fundamentally important. I want to acknowledge the funding from Flinders Foundation and Flinders University, and also the support of the Mental Health Short Stay Unit staff who are involved in understanding and working through this and helping to recruit patients. And even more important, not more importantly, the people from the SALHN Research Office and the Flinders Trial Group, and Simon's obviously here representing those, but the other groups, who helped us go through ethics, site-specific agreements, and now the ANZ clinical trials. And I believe that is the most amazing achievement that you can ever do when you get involved in these research projects. So, the context. We all know that suicide is important. We probably know someone who's had a family member or our own families where a suicide's occurred. But this is the national context. Over 3,000 people every year commit suicide, and the rate, up until COVID, ironically, the rate was going up every year for the last 5 years in Australia. Suicide rates are 3 times higher in men than women. And then the next layer is around hospitalisation due to intentional self-harm. Now, so one of our discoveries was there is no national standardised measure for suicide attempts in hospital settings. So the best you can get in ICD 10 or 11 codings is intentional self-harm admissions. And you can see where South Australia sits a little bit above the average in Australia. Now, put it in context, in southern Adelaide, we have 6,750 mental health presentations between Noarlunga and Flinders each year, in acute mental health presentations. And of those, 1,500 are coded as self-harm. So they're the ones, some of those are admitted and some of those aren't. So there's not a particular coding for actual suicide attempt. What we do know is another figure is there are about 65,000 suicide attempts each year in Australia. So in other words, roughly only probably less than half of all suicide attempts actually come to hospitals. What about our suicide rates? So this is people not in southern Adelaide as a whole, which would be twice this number, but people who have had contact in our service in the previous 12 months, either through the emergency department or for our mental health teams or inpatients. So between 15 to 20, let's say, per year. And probably the most important figure there is about 50% of those, a bit less, have been through our emergency department in the previous 6 months, and 25% of them within 4 weeks of discharge. So I'm taking you down the pathway of saying if you're really going to invest and put your efforts into preventing suicide, you target people coming into the emergency department, because that's where people are. So when we were looking at what sort of interventions we could improve our current service delivery, one of the first papers I came across was the ED SAFE in America, a team led by Ivan Miller, and they had a 5% reduction in people coming back with suicide attempts and a 30% reduction in the number of attempts. And they used what was called the class model. So that was one component. The second component is that when I read this, it was a telephone delivery system for 8 hospitals across America from one site, one telephone counselling site. And we already had the IAP service here at Flinders, which has been going for nearly 10 years, which is basically a telephone delivered service delivering brief cognitive behaviour therapy interventions. So we already had a service coming into our EDs for people who were in crisis, and we had this American model which had some similarities. So I contacted Ivan Miller, and he's been an incredibly responsive colleague now. He's part of our research team, and he provided us with manuals about how they delivered this program. And with our consumer input, we called it IAPT-M to be mobile, but actually we changed the name to My IAPT, My Access to Psychological Therapy Suicide Prevention. And we changed the name from Significant Other, mainly potentially a family member, to Trusted Other. And this was part of our whole conversation amongst our consumers and our clinicians about who is this person we're talking about, what is the core attribute that we want for this person to be partnering with our patients. We added in text messaging for the patient and the use of a Mind Tick app for the Trusted Other, and we actually produced workbooks, one for the Trusted Other, one for the participant. So there were some significant differences in the process. So the CLASS program has come on the back of Ivan Miller and his team's work for nearly 30 years in the United States around suicide prevention interventions. And it consists of the acceptance and commitment model based around values, asking someone to look at their own values, but also, interestingly enough, a family telephone intervention which came from the stroke area. So where, in other words, where people were wanting to involve the carer in the management of someone post-stroke. So they adapted that into the mental health setting. The point that CLASP is, or Ivan Miller makes, is that CLASP is not a therapeutic intervention in its own right. It's an adjunct to other treatments. When we've got workbooks, values, we talk about life plans, safety plans. The IAP provider, my app provider acts as an advisor, and we're actually targeting the suicidal ideation itself. Now, it might sound contradictory to those of you who aren't working in the mental health field, but normally we don't target the suicidal ideation directly. The way we're all trained and brought up is that you address the underlying problem. Whether it's depression, family distress or whatever, you address the underlying problem and then the suicidality will disappear. Well, it turns out there's a whole body of work saying, yes, you should address the underlying problem, but you can also address the suicidality and give people a set of tools to lead them out of suicidality. So this was actually quite a controversial discussion that we had to have amongst our own team members who balked at the idea that they were directly going to take on someone who was acutely suicidal, or potentially. So it's involved goals and it's based on people's values. I mentioned the Flinders Grant. We were funded to do a pilot program, which is overall 12 months, but the intervention will be 6 months. We aim to recruit 10 to 15 people and we monitored all of those outcomes, and at the end of it we're going to do some interviews around how the whole process went. The results so far is that of 15 people approached, 13 people agreed to be involved. 7 males out of those, 11 started their first session, 7 males, 4 females. And those ratings for depression, anxiety and disability and handicap are all in the moderate to severe range. The self-hate scale is in there as a sort of like a research understanding about mechanisms of suicidality. So self-hate is a predictor of a suicidal ideation. And where we're up to now is that of the 11 who commenced, 1 has dropped out. They've had between 3 and 6 sessions. They've been going for about 3 months. And 4 trusted others have started attending where we asked them to attend, which is a joint session at session 4. And we've started using text messaging and the Mind Tick app. I'm going to finish off with, so in other words, we haven't got any post results. We don't know what's happened in terms of people's overall scores and suicidality. We know we monitor the scoring every single session, and overall the scores on depression and those other scores are all going down, or gone back to in the normal range. And we just wanted to finish off with some quotes which Sue's generated from some of her participants. So I like that I can work through the safety plan with someone and not just be given a piece of paper to fill out with no help or support, like has happened before. This whole process has been my lifeline, and I realise I can still achieve the things I want to do, but just in a different way. Until now I thought this was all lost. And the trusted others, these sessions have helped me to start the conversation with Mum. Before she went into hospital, I just never knew she was feeling this way, and I saw her often. I feel more confident in talking with my son and asking him if he is okay without fearing to make things worse, or I don't feel bumps in the road so much anymore with my son, as I know he has the strength to manage them, and I feel like I know what to say to assist him. So the biggest lesson for me so far is about this idea and the role of the trusted other. So I was doing the recruiting of the majority of the patients, and I'd explain the basic idea to the participant. Sometimes a carer or family member was there, often they weren't. And as soon as I got to the word "and a trusted other" would be involved, there's a kind of a look came across people's face. And to a man and a woman, they all said, that's a good idea. However, when we went into it in more detail, 3 of the 11 didn't have a trusted other. Even though they were regularly visiting sometimes a family member, and I was quite specific and said, you sure you don't want someone to be your trusted other? No, I don't trust those people. And I think there's a PhD in that. That one set of statements around who the hell is your trusted other, how do you choose them and why not you choose them. The way the model works is that we can offer a substitute trusted other. So if the person doesn't want one or doesn't agree to have one or is in conflict with people, we can. And we haven't done that yet, but we've got 1 or 2 people who potentially can do that. So we think that this whole process is going to be a really powerful understanding at this stage. We can't say it's achieved any great outcomes, but Sue can ask any questions directly about how that whole works. So thank you for listening.
Thanks for another excellent talk. Are there any questions from the audience? We don't have any online at this stage.
No, it's okay.
Can I just... oh, yes, we have a question. Yeah. Microphone. Thanks.
Thank you. A great presentation, thank you. The question, even though the project's only been going for a short time, have you noticed any reduction in attendances in the ED, for example?
Not really, because it's too small a numbers.
Yeah.
So we've put in for 3 large randomised controlled trials, and we're waiting to hear so forth. And we've got a partner, our partner organisation is Royal Melbourne Hospital, and we're aiming to recruit 280 people out of 3 sites. So then I'll be able to answer that question in a bit more detail.
Thanks.
There's Michael.
Thanks, Malcolm. That's great, and a really important study too, I reckon. So is there an advantage if the supplied trusted other is a psychologist or someone like that, I guess is the first question. And the follow-up would be, has COVID made things awkward for you in this context?
Well, it's all sorts of interesting figures. I mean, we all heard about the mental health crisis produced by COVID, and that has led to an increase in presentations to hospital emergency departments across Australia, and particularly lifelines. You would have heard a lot about that. Ironically, the suicide rate has actually gone down across Australia, except in the youth area. So it's gone down into the older adults, but for 15 to 25-year-olds, it's actually gone up. So that means the sort of idea that you can deliver this sort of program completely by the phone. In other words, people remotely in South Australia or anywhere in Australia could have this kind of service. You would hope that we could actually deliver this before someone needs to come to an emergency department. And through our CBT programs in our Flinders program, postgraduate courses led by Paula Redpath and her team, we can provide this training across anywhere in Australia as well. So I think it's some great opportunities. We now already started talking about how do our own mental health staff have these skills and deliver that sort of intervention as well. One more, got time for one more question?
Just a quick question. Did you find many of your participants didn't have a trusted other? And was there ability to...
Well, I actually did mention there were 3 out of the 11 that didn't have a trusted other. And Sue worked pretty hard on trying to help them find someone or acknowledge someone. And there was a couple of times when they nearly did. But it's just, I mean, incredible conversation to have about who and why haven't you. And I suspect they are the people at highest risk of suicide. So how we can, I mean, in other words, we leave the door open that those people can come back to us. I'll better finish up. Okay.
Thank you. So our next presenter is Dr. Famida Ilyas, presenting on behalf of the investigator team, including Associate Professor Kamandi Pasquale, on their work at improving outcomes in type 2 diabetic patients with heart failure. Thank you.
Good afternoon, and thank you for this opportunity to present at SALHN Research Week. My name is Famida Ilyas, and I am the author-presenter for the clinical trial, Acute Pleiotropic Effects of Dapagliflozin in Type 2 Diabetic Patients with Heart Failure with Reduced Ejection Fraction, a crossover trial. I was involved in this trial as the Heart Failure and Research Fellow with Associate Professor Kamandi Pasquale as a principal investigator, along with the other authors listed here from Flinders Medical Center and Flinders University. Before I begin, I would like to declare that I have no disclosures. So I'll start with a brief introduction of SGLT2, or sodium glucose transporter inhibitors, and what we already know about them. So SGLT2 inhibitors function through a novel mechanism, reducing renal tubular glucose reabsorption in the proximal tubule, thereby increasing urinary glucose excretion. This results in a reduction in blood glucose without stimulating insulin release. So several large clinical trials between 2015 to 2018, such as the EMPA-REG, the CANVAS, and CVD-RAIL, have already demonstrated morbidity and mortality benefits in heart failure, in particular, reduction in worsening heart failure, including hospitalization and cardiovascular deaths. And these were diabetic trials designed to show cardiovascular outcomes as demanded by regulatory authorities for all new glucose-lowering medications. Since then, several beneficial effects of SGLT2 inhibitors have been demonstrated on different organ systems independent of glycemic control, as shown in this position paper by the Heart Failure Association of the European Society of Cardiology. This included vascular and hemodynamic, renal effects, cardiac effects, and metabolic effects. Among the benefits seen on the heart are reduced cardiac glucotoxicity, a shift in fuel supply from fatty acids to ketones that lead to improved cardiac energetics, reduction in oxidative stress, and a decrease in myocardial hypertrophy as well as fibrosis. However, the exact mechanisms of these protective effects remain speculative or unknown, and this became the basis of our research question. With larger clinical trials in heart failure with reduced ejection fraction like DAPA heart failure, EMPEROR reduced, and SOLOIST, demonstrated morbidity and mortality benefits of SGLT2 inhibitors in this cohort. The protective benefits not only remained speculative, but occurred within weeks and were independent of glucose lowering. As SGLT2 inhibitors were not supported as therapy in heart failure at the time of recruitment, we opportunistically studied the acute effects of the drug in heart failure with reduced ejection fraction, patients who had type 2 diabetes. Therefore, the aim of the clinical trial was to conduct an exploratory study using a large battery of non-invasive investigations to point to a novel and potentially acute beneficial effects of SGLT2 inhibitors in heart failure with reduced ejection fraction patients with type 2 diabetes. And the parameters studied were comprehensive yet not exhaustive and included functional capacity assessed through the 6 minutes walk test and question of life score, clinical status by means of physical examination and New York Heart Association classification, hemodynamics through 24-hour blood pressure and radial artery tonography, echocardiography and lung ultrasound, electrophysiology parameters including ventricular ectopic burden and heart rate variability, as well as biochemical measures of hematological, renal, liver, and cardiac markers. For the methods, the trial received approval from the Southern Adelaide Clinical Human Research Ethics Committee. It was partially supported by a donated funding grant through Flinders Medical Center Foundation. A family had left behind money for a cardiology research project, and I would like to thank them and acknowledge their generosity here. The study drug was dapagliflozin, and it was, as well as its matching placebo, supplied by AstraZeneca. Ambulatory blood pressure and Holter monitor equipment was supplied by Flinders Cardiac, Heart and Vascular. And for the statistics, the results were displayed as a median or number percentage and compared with Wilcoxon-signed rank test for continuous variables. It was designed as a randomized double-blind placebo-controlled crossover trial. It was exploratory in nature, as mechanisms for beneficial effects of SGLT2 inhibitors remain not clearly defined. So for the study design, the trial was powered to show a 20% difference in the 6 minutes walk test, and the number to achieve this was 22 patients. 25 subjects were selected to account for potential dropouts. However, the study was stopped at 19 subjects following the results of a large study, the Imperial trial, that showed no significant benefit in 6 minutes walk tests. So at the end, we had 24 type 2 diabetes patients with heart failure with reduced ejection fraction of less than 40% who are eligible to receive SGLT2 inhibitors as add-on therapy for their diabetes according to current guidelines, identified and screened. They were at least 2 weeks away from hospitalization for decompensated heart failure prior to randomization, and were stabilized on maximally tolerated doses of guideline-directed medical therapy for heart failure. After baseline investigations, subjects were then randomized to either receive 10 milligrams daily of dapagliflozin or its matching placebo, and after 2 weeks, the tests were repeated. Following that, they went through a washout period for 2 weeks, and then were crossed over for another 2 weeks, and the final tests were then repeated. All patients at the end of the trial received open-label SGLT2 inhibitors. So we go to the results, and we first look at the 6 minutes walk test, where there was no significant change in distance after 2 weeks, and this is what was confirmed by the other larger trials like the Imperial trial. The difference here being calculated between baseline placebo and baseline dapagliflozin. Next, and I apologize for the big table, that shows the results of clinical and functional hemodynamic and electrophysiology parameters at baseline and after 2 weeks of placebo and dapagliflozin. The parameters in red demonstrating statistical difference. First, dapagliflozin had no significant effect on clinical, functional, or quality of life parameters, except for body weight, where it fell marginally. Dapagliflozin reduced systolic and diastolic blood pressures, and this was a relatively large reduction that we saw. Radial artery-derived central arterial pulse pressure fell. However, peripheral capillary-derived reactive hyperemia index, which basically measures the microvascular endothelial function, unexpectedly fell. Dapagliflozin did not change the average heart rate. However, there was a reduction in the burden of ventricular ectopes as well as heart rate variability. However, results for imaging parameters showed no effect on cardiac chamber size, ventricular systolic function, or lung ultrasound detection of interstitial lung water. We were able to demonstrate some change in biochemical parameters. First, there was an increase in serum hemoglobin, creatinine, and albumin. And this supported the evidence for diuresis and hemoconcentration. There was increased high sensitivity troponin T, and this was somewhat unexpected for us. However, there was no change in NT-proBNP. Summarizing these results for discussion, after 2 weeks in 19 patients, there were early beneficial effects that were expected and consistent with larger clinical trials, but there were 2 changes that were not expected. The reduction in microvascular endothelial function, which was measured through reactive hyperemic index, as well as increased high sensitivity troponin T, and these 2 warrant further exploration. There was no change in functional or clinical parameters except a small change in weight loss. A relatively large blood pressure reduction was demonstrated with evidence of reduced central arterial stiffness. A reduced arrhythmic propensity was supported by reduced ventricular ectopic burden. However, we did not see any reduction in interstitial lung water, NT-proBNP, or echocardiographic changes to support reduced filling pressures. And so this really went against the idea that we could reduce congestion with just acute simple diuresis during the immediate period. In conclusion, in a cohort of 19 patients at 2 weeks therapy, several parameters had changed, with some were expected on proposed mechanisms and some were not. We acknowledge the small number of trial, despite the crossover design that increases the trial power to show through effects. However, the number of changes that we found suggested multiple mechanisms of action, and this would be hypothesis generating, so that we could focus on where future studies should concentrate on in heart failure with reduced ejection fraction. Another addition that I would like to add is that since we've done this clinical trial, we have found that it has had clinical impact, and it has changed the way we've managed our patients with heart failure with reduced ejection fraction. The first point here I would like to make is the fact that since we have demonstrated a large reduction in blood pressure, we now know that when we manage patients who are hypertensive and who have heart failure with reduced ejection fraction or diabetic patients, initiating SGLT2 inhibitors will help reduce their blood pressure. But also, has a cautionary advice, we know that in the cohort of patients, say the geriatric patients or patients on multiple antihypertensives, we do need to watch for adverse events, given that it does lower blood pressure here. At the moment, SGLT2 inhibitors are not marketed as blood pressure lowering medication, and therefore these effects on blood pressure will not be able to be advertised. So it is important for us to recognize that this is either a good effect or a side effect, a salutary effect, or not such a good effect in certain patients. The second point I'd like to make here is that we now realize that there are good benefits in initiating dapagliflozin in the immediate period after heart failure. Traditionally, we would start novel anti-heart failure medications 3 to 6 months or even a period beyond that. However, we now propose and believe that we should initiate SGLT2 inhibitors much earlier, and probably propose that they should be initiated while in hospital at a period of when they've been stabilized from their decompensation and prior to being discharged from the hospital. Last but not least, we are quite excited that this clinical trial has been published in the European Society of Cardiology Heart Failure Journal in August this year. Thank you.
Thank you for another excellent presentation. We don't at this point have any questions online. Are there any questions for Dr. Ilias from the audience? While people are just thinking, could I ask a question? You mentioned that there is perhaps this side effect of changing practice around treatment in this group of patients. Do you think, based on evidence from your trial that has been published and also sort of surrounding evidence from others, that this is warranted, or is this in fact perhaps a little bit too soon, which sometimes does happen in clinical practice?
Thank you for the question. I think it is warranted. I think we are going to see, in fact, I'm going to probably say that there's so much more trials going on with the other systems, like we've got DAPA-CKD in the renal patients, and I think we're going to see all very similar effects, which actually we are quite excited about because we think it's actually going to prove more beneficial for our patients. But obviously, just like any sort of medical therapy or management that we do, there isn't a cookie cutter sort of approach. We do need to be aware about those cohort of patients who there will be adverse events related to it. But I guess the first thing is that by showing these immediate effects, or by understanding these mechanisms, which is really what this trial was designed for and our desire, when we understand and we have the knowledge behind it, then we are better able to designate a certain management for different patients.
Okay, thanks. We have one more question. Sorry about that.
Sorry to pull you back. I just want to talk about the reactive hyperemia, which I thought was a bit interesting there. And many years ago, reactive hyperemia was thought maybe to be reduced in heart failure associated with edema at least. And so if you had reduced edema, you should see improved reactive hyperemia. So I think you showed us that there was a loss of weight and maybe the albumin went up, which would be consistent with perhaps a reduction in water.
That's right, diurescence, yeah.
And you might have expected an improvement in reactive hyperemia, but you had the opposite effect. So can you help us understand that a little bit?
Absolutely. So, and that is where I said that was unexpected, because we would have expected the opposite of it. I guess the one, and we tried to sort of discuss around why we thought this could have happened. I think there is also, and sometimes what we see in the coronary arteries in particular, is that when we saw central arterial blood pressure reduction, and we wonder whether downstream, where you've got the microvascular capillaries and vessels, if there's a reduction in the central blood pressure, to maintain perfusion, whether they undergo vasoconstriction rather than vasodilatation. And that was, I guess, the only thing we could come about. Again, we didn't have enough to probably say, yes, that is probably one of the mechanisms, but definitely it was unexpected as well for us. And we felt that our trial numbers are quite small. We probably would not be able to explain it and justify it, and that warranted further exploration. In fact, with on the discussion of the reduction in hemoglobin, creatinine, and body weight, we thought that, yes, that's all to do with diuresis and hemoconcentration. But having said that, we do know, especially I think with the DAPA-CKD trials and in the renal trials, which has just been published, and I'm sure we're going to start discussing about it, we do think there might be renal effects as well affecting all these biochemical parameters. So the weight reduction was marginal. We do know that erythropoietin is stimulated by SGLT2 inhibitors, and that can cause increase in hemoglobin concentrations. We do know that there is a tubuloglomerular feedback with natriuresis from SGLT2 inhibitors, and that in turn leads to afferent arterial vasoconstriction and hyperinfiltration, and therefore that can lead to the rise in serum creatinine that we see in the initial initiation of dapagliflozins or SGLT2 inhibitors. What we all know has a reversible dip that occurs within the first 4 weeks. So a lot of the biochemical parameters that we saw, we could actually explain by what we are seeing and by, diuresis, hemoconcentration, but we think there's also the renal effects and other endocrine effects that there's a much larger sort of mechanism that's all working together synergistically to cause these changes. Thank you.
Thank you. I'd like to invite our next speaker to present. So Dr. Shailesh Bahari presenting on induced hypernatremia in patients with moderate to severe ARDS. Thank you.
Thank you very much. I'm presenting on behalf of my team, all mentioned on the slide there. This paper was recently published in Intensive Care Medicine Experimental, and I'm going to go through this study in a stepwise fashion. But it's all about ARDS patient, which is very common in intensive care. They have a high mortality, and there's no real no therapy for them. We manage them in a conservative way on a ventilator. And this we're talking about induced hypernatremia. Now, people who are aware of this area will surprise with this, because hypernatremia across the board in any cohort of patient in the hospital leads to increased mortality. This has been shown in many times again, over and over again. Either you come with hypernatremia or you develop hypernatremia in the hospital setting, you have increased mortality. Doesn't make sense, does it? But if you look from the basic mechanism point of view, there is a lot of data. Now, this is hyperosmolarity. I'll come to it. Both from the cell side of things, from the channel side of things, from the modulating of gene expression to various agents which can modulate attraction of neutrophils, adhesion molecules. This seems to be suppressed with the induction of hyperosmolarity. Now, hyperosmolarity essentially in these models were used by sucrose, and safety of sucrose in humans is not known, and probably we know that there's a lot of concerns about using colloids, synthetic colloids. But osmolarity essentially is sodium, glucose, and urea in a general sense of way, and more than 95% of osmolarity is sodium. And we use hypernatremia all the time in traumatic brain injury, for example, and we got a lot of clinical experience for it. And it's quite simple, really, to increase serum sodium in patients in the intensive care unit, and it's not been tested before. Now, why ARDS? I've just put a slide here for people who are not aware of ARDS. ARDS, acute respiratory distress syndrome. It's quite common. It is made of many diagnoses. These patients are quite sick on a ventilator. And the mortality rate, this is the best data we have in a moderate to severe ARDS, is roughly around 35% to 40%. So I'm talking about a very sick cohort of patient in intensive care unit. Now, in the spirit of what we've been doing this week, we actually did some basic science work before we embarked on the clinical trial of some animal work. This is a part of my PhD, where in a lung lipopolysaccharide model, rat model, in a lung injury model, we showed that it was protective in those animals, and we went through various mechanistic pathways, how it might work. This probably is outside the scope of today's talk. And in the spirit of tomorrow's day, where there's all large databases stuff, we actually went and looked at the ANZEX core database and looked at the effect of hyponatremia and actually tried to understand it a bit further. And after we actually went, this is over 1 million patient data from the core database, and we found that there is a differential effect. Okay, the hyponatremia actually increases mortality across the board, no doubt, except in patients with possible lung injury, where there was an amelioration of the expected increase in mortality. This is essentially the basis for the hypothesis we had, that if we increase the sodium in patients with lung injury from to around 145 to 150 in moderate to severe ARDS, it will reduce, it is feasible, first of all, and it will reduce the severity of lung injury. So we got ethics approval for this. These trials were registered, and we got funding from Intensive Care Foundation in Australia to run this trial. So it was essentially a prospective, randomized, proof of concept, single-center study, where we included patients with ARDS who were adult and within 48 hours of diagnosis, so quite early on during the journey. We had a bunch of exclusion criteria, but more importantly, we had patients with traumatic brain injury because it's used as a therapy for traumatic brain injury. We excluded them. Now, sample size was hard to calculate. We estimate the sample size based on a previous trial by Meduri et al., who was a big name in this ARDS research. And we used their primary outcome as a physiological outcome and used a similar methodology, sample size calculation. We estimated on 20 subjects per group. So the primary endpoint was, now this is a both clinical and physiological endpoint, which was a 1-point reduction in lung injury score. Or you are successfully extubated, that means you're not on a breathing machine by day 7. What is lung injury score? It's a composite score of various things, which had chest radiograph, had hypoxemia, that's how much oxygen and what's your oxygen levels in the blood. Your amount of PEEP you're requiring on the ventilator. PEEP is the pressure which you give when you are breathing out. And your respiratory system compliance. So it's a composite outcome of these 4 parameters. And we had multiple secondary outcomes, and we'll go through them in a second, but we had ventilator-free days, chest x-rays, lung injury scores, length of ventilation, and then ICU and hospital outcomes. So essentially what we're doing was, in the intervention group, we are trying to maintain the sodium between 145 and 150. We are managing them as the expected management in an intensive care unit with mostly conservative fluid management and lung protective mechanical ventilation. This is the protocol we followed, where we manage the sodium between 145 and 150. It's a busy slide, but I will just mention how we actually did this. And we compared them with a conservative fluid management strategy and a lung protective ventilation strategy. This is a consort diagram. We assessed around 101 patients for eligibility. There were significant exclusions, and some of the exclusions were around consent, or we were too late in getting to the patient. They were already 48 hours in the unit. In the end, we had 20 patients in each group, which we followed up during the duration of the study. Baseline characteristics were well matched between the groups. I won't spend too much time into it, but there was no significant difference between the baseline characteristics. First of all, let's come to the results. So question number 1, did the protocol work? So there was a significant difference in the sodium levels and the chloride levels in the intervention arm. So the first question was answered, it was feasible. Second question, is it safe? So we looked at various parameters or surrogates of safety. This is cumulative fluid balance. From an intensive care perspective, fluid balance is very important, and a liter excess of fluid balance has mortality implications. And there was no difference in the fluid balance. SOFA scores is an organ failure assessment score. The organ failure recovery rate were same between the 2 groups. There is a school of thought that chloride levels can affect your kidney functions. Hence, we looked at the kidney levels, kidney functions carefully, from the routine ways of measuring it. And there was no difference in the urea and the creatinine levels. All right, coming to the main findings. The primary outcome was a 1-point reduction in lung injury score or successful extubation in day 1 was significantly better in favor of the hypertonic saline group. There were other bunch of secondary outcomes which had length of ventilation or hospital length of stay were also in the favor of hypertonic saline group. This table also looked at various use of renal replacement therapy or diuretic use, inhaled nitric oxide. There was no difference. And also gives an opportunity for sample size calculation for future trials, which I'll talk about in a second. There are a bunch of secondary outcomes. There are much more slides after the end slide here. But essentially, they were either in benefit, favor of hypertonic saline, or were not different between the groups. We had multiple exploratory outcomes. We actually collected blood samples and bronchial lavage in some of these patients, not all. Quite little disappointed. There wasn't any difference between them, but there was a lot of baseline difference. And essentially, in a sample size, that's quite possible. We are trying to look at the mechanistic angle. If you look at the plasma angiopoietin 2 or the BAL TNF alpha, there was a lot of baseline difference, which we think accounted for the lack of difference overall. All right. So main finding, this was the first prospective randomized study to demonstrate a reduction in lung injury and earlier extubation in patients with moderate and severe ARDS. It's not only a feasible intervention, appears safe, and in whatever ways we measured it. There are multiple limitations in the study. Obviously, it's a small size, single center. We have seen this in multiple times in research with lung injury, that a single center study may not be replicated in multiple centers, so that has to be replicated for adequately powered patient-centered outcomes. So we did some calculations. If you make the primary outcome as ventilation-free days at 28 days, which is quite a standard outcome, we'll need 142 patients per group based on the data we had. We probably need more robust markers of kidney function. I mean, we use creatinine and urea, but probably there are much more markers available, which should be utilized in future studies. Long-term cognitive outcomes, so there is a lot of data out there where conservative fluid balance strategy can lead to a cognitive decline in patients in future, and that's something which needs to be looked at in this study. We didn't find any difference in plasma and BAL, which is different from our animal study. But so we can't really find insight into the mechanism, but there was a lot of baseline differences, and this should be a part of the next phase 2 trial. Now, part of my current research work is to look at inflammatory phenotypes of ARDS, and perhaps that should be integrated into this future research. All right, so to conclude, induced hyponatremia is commonly done in ICU for a lot of patients with a brain injury problem. It can be done in moderate to severe ARDS patient. We can use hypertonic saline. It's feasible, and it does lead to resolution of lung injury in early extubation. Perhaps a larger phase 2 trial is now warranted to look at this and understand the mechanism behind this. Now, thank you, and thank you to all the researchers involved in this research. We are very excited to finish this because it started as a basic research question, went to a large database, and then we're finishing the first stage of the clinical trial. I also want to thank all the ICU staff who were involved or indirectly involved, all the nursing staff. But perhaps the most important thank you I want to give is the relative or next of kin of those patients who cannot give consent for these trials. And essentially, this is a very experimental trial. To give a consent for these kind of studies is hard. And I want to thank all the relatives and the next of kin who became a surrogate and gave consent for this study. Thank you.
Thank you. Are there any questions from the audience at this time? We don't have any questions online at the moment while people are perhaps thinking. I wonder, here we go. Yes, please, go ahead.
2 questions, if I may. The first is, thank you. Given that we know that people die faster in the hospitals when their serum sodium goes up, did you have much difficulty with getting this through ethics? That's the first question. The second question is, you went through it pretty fast there, but there seemed to be a massive difference in the length of stay of the 2 groups. Massive difference in the length of stay. It's 19 days versus 46 or so. How do you explain that?
Yeah, so the first question is ethics. Essentially, because we had so much of data behind this, both from the basic science perspective and large databases, that it was not difficult, but reasonable from what we should have been. So there were multiple checkpoints in this. This is a proof of concept phase 2A trial, so there was a reason to stop the study early if we find that this trial was not feasible, for example. So that's one. Now, in terms of your length of stay data, I think basic thing in this is length of stay in the ICU and the hospital is heavily relevant on the number of time you're on the ventilator. The longer you're on a ventilator, you get weaker and weaker. You get chances of neuromuscular weakness. You get chances of all the infection problems. So length of stay will always be in favor. A single day reduction in the mechanical ventilation will have a huge impact on total length of stay.
So you think that's a real result, 19 days versus 46 days?
Correct. So in patients who are on a breathing machine will require tracheostomy more than a week per se, and tracheostomy means they will be staying in ICU for at least, many weeks.
We do have one question online. It's probably a question that's bigger than 1 or 2 minutes, but maybe if you could just summarise briefly. The question is, what are the challenges conducting clinical trials in ICU? You've perhaps already alluded to some of them around the consent, but maybe if you could summarise a few others for the audience. Thank you.
Okay, that's a broad concept question. I suppose there are many challenges. Let's talk from the first principle. Is the question you're asking, is it relevant to the patient's condition? Because patients have multifactorial problems in the intensive care unit. To find a one bullet to treat everyone is hard. I think that's number one, and that's been the major problem with ARDS research, and that's why I mentioned now we are going into various inflammatory subtypes where we can perhaps, have much more targeted therapy. Second is there is a time clock ticking, right, ticking in the background. Most of these trials will require patients to be enrolled within a certain time period, otherwise it's too late. And that too late means there is challenges. If there's a weekend admission, there's overnight admissions, there are admissions at a time when you're not around. So there's a time ticking when in these trials. Third, obviously, most of the patients cannot give consent. So consent becomes quite a challenging thing. The way we follow these consent is that either we take a permission from the patient when he's better, or the next of kin has the option to withdraw from this. Now, Australia is unique in a way in many countries, because some of these, not in this trial because you needed consent, this was quite a very early in its inception as a concept to study. But many trials do give you permission to enroll without taking consent and then go and take consent within 24 hours with the next of kin. There is an option of taking phone consents now, because someone comes after cardiac shock and is on 40 of vasopressors. By the time you find someone to take consent and stuff and use a drug which may be of some benefit or may not be of some benefit, we don't know it, the time might have gone. So that's there, and that's something ethics and the governance has been looking into. Every state has a different jurisdiction, for example. For example, this is not possible in New Zealand. Third is, all right, and this is very relevant to your point, and that point is about what outcomes you're looking at. So what if they did well? 6-month mortality is not different, right? It's the outcome thing, and that has various layers to it. I still believe a day saved on a ventilator is a very relevant outcome on a patient, just from the suffering they go through, the sedation they go through, the infection risk they go through, the amount of, from the hospital point of view, the amount of finance required to look after a patient on a ventilator is around $5,000 a day. So if you save one day on a ventilator, you save around $5,000 a day per patient. Now, then there is long-term outcome. Mortality actually is coming down, I don't know, not through one intervention, through multiple interventions. And whether is there actually the health quality is getting better, or 1 year mortality is still the same, or there's something like neurocognitive outcomes. Maybe if you save them, especially it's very relevant to cardiac arrest studies, it's not about saving them, it's about what their functionality is in 6 months. So I think there is a moving kind of a area which a lot of work has been done in this area. So I think that's some of the challenges I can see.
Yeah, great, thank you. Thanks.
Thank you.
Thank you. Another excellent presentation, and interesting to hear about some of the great research, but also the challenges happening here at SALHN in clinical trials. So our second to last speaker is Dr. Tamina Levy, and she's going to talk to us about work around upper limb activity in chronic stroke. Let's get through these for you. Keep going.
Yeah,
There
we go. Thank you. Hello, everybody. I might just start by saying I hope I'm semi-coherent. I've actually been moving house today, so it's been a big day already. And I'd like to thank Research Week for accepting my presentation and for putting me on with all these amazing professors. What an incredible honour. So I'm going to talk today about the INTENSE trial. The INTENSE trial, which stands for the Intensive Therapy Efficacy After Neurological Spasticity Treatment. My role within this trial was as one of the senior therapists in delivering the intervention, but I was also involved in the development, implementation and analysis of the pilot study which happened before the INTENSE trial. And following the INTENSE trial, as a component of my PhD exploring exercise adherence in stroke survivors, I did a qualitative analysis of the study. So I'm going to touch on that towards the end. So we know that stroke survivors, many of them have difficulty moving their arm following stroke, and this is a key reason for them failing to meet their treatment goals. And one of the main reasons for that can be increased spasticity in their upper limb. One of the treatments for spasticity that we use is botulinum toxin A injections. People commonly refer to that as Botox. Botox is a brand name of botulinum toxin A. And whilst there's synthesised open-label evidence that botulinum toxin injections reduce spasticity when compared to placebo, there's no evidence that it leads to improved function. But in spite of this, many of the clinical guidelines we have still recommend that people with moderate or more spasticity should have botulinum toxin A injections followed by therapy. However, that's really based on expert opinion rather than sound evidence. So we chose to test this out in a clinical trial, and the trial aimed to determine the clinical effect and the cost-benefit of adding intensive upper limb therapy to botulinum toxin A injections. So the actual question was, in stroke survivors attending a spasticity clinic who are scheduled to receive toxin, is adding 3 months of evidence-based movement training more effective than providing a simple handout for exercises in terms of goal attainment, scaling and upper limb activity? People were eligible for this study if they were more than 3 months following stroke. They could not have had toxin within the 6 months leading up to it. And they needed to be eligible or scheduled to be having botulinum toxin injections to muscles that cross the wrist. So this was a NH and MRC funded multi-centre trial, and REPAT and subsequently Flinders Rat were actually the main recruiter for this study. So there was concealed randomisation, and that occurred following injection. So everyone in the trial received their botulinum toxin injections, and then they were randomised following that into either just a generic handout of exercises or the intensive protocol delivered through the INTENSE program. So what the INTENSE program looked like was, it was a 12-week program, so it was a really big commitment for people. It started with 2 weeks of serial casting. So basically, most people had injections into muscles that cross the wrist, so they were presenting with spasticity. And then we would put them we'd stretch them out and put them into a cast at maximal wrist extension. So for me, that's back there, but for a lot of our patients who are in a contracted position, that we would stretch them as far as we could and put them into a cast. And they usually had 3 casts over 2 weeks. As soon as the cast came on, they started their 10 weeks of intensive motor retraining. And that was a big commitment. They were asked to do an hour of practice every day. It was largely patient-driven, so a lot of it was based at home. An hour of practice every day for over 10 weeks, so 70 hours of practice. So it required a really big commitment from people. So you would think they were generally motivated, quite motivated types of people. The exercises were, so following the casting, they started this motor retraining. There was sort of 4 different levels of exercises. So if people didn't have any ability to move their hand, they would use an electrical stimulation machine to try and facilitate some activity. And then that progressed to the level 3, who were actually able to do quite a lot, and they might have even had little weights to work their hand. So it was sort of individualised, based on a program called the GRASP program, but very individualised for that patient. So the home-based program every day was supplemented by visits into the clinic, and that started sort of a couple of times a week, and it reduced throughout that period. They also had a home visit to set them up and make sure that their environment was conducive to the exercise program, and then they had some phone calls just to check in on how they were going. The control group at the time of injection just received a very generic exercise sheet with some upper limb stretches and exercises for them to do. Both groups were asked to keep a training log, so to record how long they were exercising for every day. And for the intervention stream, there was also an audit of clinical sessions, so each clinic throughout Australia had an audit to make sure people were adhering to the protocol. It was a very strict protocol. So looking at the flow of participants through the trial, so there was 7 clinics through Australia, and after screening there was 140 participants, and that met the target recommendation which was developed from the pilot study. So it was a fully powered trial. Baseline measures occurred before they had their injections, so for both groups. And the primary outcome measures were the goal attainment scaling, which is an individualised outcome measure of setting a specific goal and giving it a score. And it's used a lot in spasticity research. And the box and block test, which was measuring upper limb activity, where there's basically a box with a partition in the middle, and there's a whole lot of blocks on one side, and the person is asked to pick up a block and move it over the other side. And they have a minute to do that, and you count how many blocks they do. So that's a very standardised measure to look at. There were some secondary measures taken as well, including strength, pain, burden of care, and quality of life. So looking at the participants, key demographics were very similar. Really interesting, mean time since stroke was more than 3 years. So they were a much more chronic group than we were expecting, and they were actually a more chronic group in the pilot study. And I think this probably had an influence on the results. And if you look at the box and block score, the mean was less than 1, so they were really impaired. So they were quite far down the track as far as their stroke journey, but they also had a lot of impairment, so many of them couldn't move their hand at all. So did it work? Well, the short answer is no. So despite this intensity, the key outcome measures at 3 months, the gas and the box and block test, there was no significant difference. So in terms of the gas, the difference was 2, but minimal detectable change in relation to gas T-scores is said to be 10, so that was actually way off. And as well as no difference in these primary outcome measures, there was the only secondary outcome measure there was a difference was strength, and that was in favour of the experimental group. They did have increase in strength, but that actually didn't then lead to an improved function as measured on the gas goals. So in summary, this RCT found that in a group of chronic stroke survivors, adding 3 months of evidence-based training was not more effective than just giving them a handout. So that was a bit of a shock, because we sort of were seeing this as a real trial that would help justify some of the resources we were at the time using. So why? Well, there's been a lot of discussion about this, and really I think it probably relates to the fact that these patients were extremely chronic and very, very impaired. So there's a belief that probably this trial came too late for a lot of them, and if it had been earlier in their journey, things might have been different. So they were very chronic. The mean box and block was only 2 blocks compared to 70 in age match. So it's a real difference. And 75% of them had 0. So 75% of the cohort could not actually pick up a block and move it over to the other side in a minute. So, yeah. And we did find that the higher level patients who were the responders actually did show some change in strength. So maybe if we caught people earlier, we actually might have made a difference and got different outcomes. On a positive note, this protocol was highly effective at getting people to practice, and in fact their adherence rate was reported even at the end of the 3 months to be at 62%, which is actually really high for a 3-month study. So that was really interesting. So why? These people weren't getting better, but they kept going and they kept practicing, many of them. And so we thought that would be really interesting to explore as a little bit of an addition to the RCT. So that's where we looked at the qualitative study. So what we're wanting to do was to basically determine, well, what drove that? What made them keep going, even though they actually weren't really getting better? So we wanted to explore what the barriers and enablers to practice were. So we followed a fairly standardised and rigorous process for qualitative research and interviewed 20 participants from the intervention stream of the INTENSE trial. We coded our findings using the COM-B model, which is a behaviour change model which is used extensively in the adherence literature. Within the COM-B model, it says that capability, opportunity and motivation are the three main factors which drive our behaviour. And if you probably think about your own behaviour and going to the gym, things like that, you can think about how those three main factors all interact. So it was a really sound model to base this study on. So what did we find? Some of the key barriers. Even though these exercises had been tailored for these patients individually, so many of them just said they were just too hard. And that led to other barriers, such as people getting frustrated. So some of those quotes, I get frustrated because I just can't do it. I know my hand won't do it. And I think, well, why should I do it? And then there were some of the more typical stroke-type barriers, such as pain and fatigue, that, if it hurt, people just didn't want to do it. Feeling frustrated was another one that linked into being too hard. And relying on others. People didn't really like the fact that they might need a carer to help set them up with their exercises. Some of the enablers were really important to us, particularly as clinicians, when we're prescribing exercises for stroke survivors. The importance of social support. For most of them, there was one gentleman that always exercised when his wife wasn't there because he said that she always told him he was doing it wrong. But almost everyone else said they really valued someone there to motivate them, to remind them, to help them set up the exercises. The relationship with the therapist was so important, particularly the face-to-face contact. So that's a bit of a challenge for us with more telehealth these days. The sense of accountability. I said I'd do it. I gave you my word. I'm not going back on my word. So even though I'm not getting better, I'm going to keep going. And I guess the overriding message was hope. So people said, I know, I wasn't getting better, but I just kept thinking, if I just keep going, something's going to change. And I think that, again, for us as clinicians is really important because we often think, we worry about giving people false hope. But if we actually don't let people hope, then they may not actually be motivated to do anything. So that's really important. So the clinical message is a sense of hope is important, therapists play a critical role, and consideration must be given to the timing and tailoring of intensive exercise programs. And that's all. Thank you.
Thank you. Lovely to see the quantitative and qualitative threads of research there linking together really nicely. Are there any questions from the audience? And we have time for one question. We don't have any questions online at the moment. One question down here.
Yep. You seem to spend a lot of time reflecting on why. You got the result that you got, as though you were implying that it was not true.
No, it was just a surprise. It wasn't what we were expecting.
But it may well have been a true result.
Yeah, it was a true result.
Okay, so all of this saying, we should have done it earlier, etc., etc.
I didn't mean to imply it wasn't true. It was just not what we sort of thought this was going to be the trial that showed this is what we needed to do because this is what we were doing. Yeah. And I think we were surprised just at the complete lack of significance in the results, despite this enormous amount of resources that these people were getting.
Yeah, you can save some money now.
Yeah. Well, this trial certainly said that for this group, we really can't justify that kind of intensity and amount of resources. Yeah.
Great.
Thank you.
Okay, thank you. So we'll move on to our last speaker. So Dr. Angela Chen is going to speak about clinical factors that affect insulin dose requirements during treatment of prednisolone-induced hypoglycemia. Thank you, Angela.
Cool. Right. Good afternoon, and I'll try to not keep people here any later than they have to be, but thank you very much to the organising committee for the opportunity to present this work on behalf of the team. I'm a PhD candidate at the School of Medicine here at Flinders University. I have no disclosures. So I don't really need to explain to this audience that inpatient glucocorticoids are used therapeutically in a large number of hospitalised patients, and specifically prednisolone is prescribed to up to 25% of all patients who are in hospital. Of this group of patients who are prescribed a short burst of high-dose glucocorticoid, a large number of them experience hypoglycemia, and it's well known that new inpatient hypoglycemia is associated with many adverse effects, including increased mortality, morbidity, and prolonged hospitalisation. So many international societies have actually released guidelines to try to inform management of glucocorticoid-induced hypoglycemia, and specifically, the Australian Diabetes Society, Endocrine Society, and some British societies as well. There's kind of two main messages that run through these recommendations, and these are firstly that all patients should be treated with insulin, and secondly that this insulin dose should be weight-based, so ranging between 0.1 to 0.5 units per kilo have been the current recommendations. So what we have previously shown is that even when you do treat these patients with a weight-based insulin dose, based on these recommendations, that a large proportion of patients still aren't meeting these glycemic targets. So if you look at panel A, you'll see that regardless of the basal insulin used, a large proportion of patients still are outside of the target blood glucose range. And then if you look over on panel B, the mean glucose concentration in these groups, who by and large have been prescribed 0.5 units per kilo, is still about 10 millimoles per litre. And then when we looked at this data further, what we found was that with this weight-based insulin dose, it was interesting to see that a significant proportion or a large proportion of participants were either always within the target range, but equally a very large number of participants or patients were always constantly outside of this target blood glucose range. So when you look at this, you think, well, there must be other clinical or there must be other variables at play beyond just the weight itself, and we should probably take these into consideration. So the aim, therefore, of the current study was to define actually the clinical features associated with daily insulin requirements in these hospitalised patients who are treated with prednisolone and then subsequently experience hypoglycemia as a result of this. And our expected outcome was that incorporation of these other clinical variables into an insulin dosing algorithm will hopefully help us improve these glycemic outcomes in this specific patient cohort. So in order to achieve this, we conducted a prospective interventional study. This involved 50 adult medical inpatients who are older than 18 years and prescribed a single morning dose of prednisolone of 20 milligrams or more, and they had to have hypoglycemia as defined by either 1 blood glucose greater than 15 or 2 greater than 10 millimoles per litre. And then in terms of the intervention to actually calculate their 24-hour insulin requirements, all patients were placed on an intravenous ActRapid infusion consisting of 50 units of ActRapid in 50 mils of normal saline. And then the rate of the insulin infusion was adjusted on an hourly basis based on glucose measurements from a flash glucose sensor. And then we performed univariate and multivariate linear regression to try to examine these relationships. So this is how recruitment flowed. Between July 2019 to July 2021, there were 148 potential participants identified on the wards that met this inclusion criteria. Of those, 98 weren't admitted, so 50 patients were prescribed an insulin infusion, and then of those, 3 withdrew to the reasons outlined here. So the final analysis involved 47 participants in this study. In terms of the baseline patient characteristics, this is not very typical for your general medical inpatient ward. So mean age was 69 years. we had more men than women. Weight was 94 kilos, and the average BMI of the participants put them into the obese range. Admission diagnosis, so the vast majority of these patients were admitted with a respiratory diagnosis, which is not unusual considering the therapy of prednisolone that was used. And large number of these participants had known diabetes already, and this is reflected in the HbA1c of 7.7%. And the peak glucose, or the mean peak glucose on the day prior to commencing insulin infusion was 16.5 millimoles per litre. 30% of patients weren't on any diet form of treatment for their diabetes at all, or for any form of diabetes treatment at all, and then 25% of participants had already received diabetes treatment to the point of requiring insulin. So this is their daily glucose profile. It follows that is typical of prednisolone-induced hypoglycemia. Mean glucose that was achieved on the insulin infusion was 8.1 millimoles per litre. And this is their insulin requirement profile, which follows that of their mean glucose profile, and mean insulin requirement was 2.8 units per hour. So we then adjusted their daily 24-hour total daily insulin to target a glucose target of 8 millimoles per litre. I'm just going to show how we did these calculations. We chose 8 simply because that is the middle of the range that's recommended across a range of societies. In terms of, so we looked at the slope between the relationship between mean glucose there on the x-axis and plotted that against the raw insulin dose on the y-axis, and that had a strong linear regression, and that gave us a slope of 16.4, and that was put into this formula. And then to kind of demonstrate how we adjusted these doses, so take this participant in the blue. Their mean glucose concentration over 24 hours was 6.4 millimoles per litre, and they used 31 units of insulin. So in order to target a blood glucose of 8 millimoles per litre, they'd require less insulin. So our calculations suggested that they would only require 5 units per day, whereas in comparison, this patient here in the yellow, so their mean glucose concentration was 11.6 millimoles per litre over 24 hours, so they would require more insulin to target a mean glucose of 8 millimoles per litre, and this was adjusted to 183 units per day. So following on from there, this was the univariate relationship between their calculated daily insulin dose and their weight, which is the main thing that we use now. And certainly that association is significant, but the R-square valued here is 0.11 with quite a large amount of scatter. Then we looked at some of the other predictors of their daily insulin dose, both in univariate and multivariate analyses, and just walking you through the univariate analyses. So what we found in terms of significant predictors of daily insulin requirement in this group was sex was one of them, patient weight was certainly one of them. Interestingly, illness factors such as the diagnosis on admission, what the average prednisolone dose that they used, a CRP as a surrogate marker of inflammation, none of these predicted for their daily insulin requirement. However, glycemic factors including baseline HbA1c, their peak glucose and diabetes treatment certainly did predict for their 24-hour insulin dose requirements. When this was then analysed in multivariate analysis, what we found was that the factors that were significant once again were sex and weight, and then when we looked at the glycemic factors, HbA1c continued to be significant, whether the patient had diabetes or not and their diabetes treatment, but certainly peak glucose was no longer a significant predictor. When we looked at this in a model then and actually looked at these multiple variables and put them into multivariate analysis, what we found was incorporating 5 clinical factors that included, in addition to weight, which we already use, so incorporating other easily measurable factors including HbA1c, accounting patient sex, diabetes and their diabetes status and their diabetes treatment could account for up to 65% of the variability in terms of how much insulin a patient requires for treatment of prednisolone-induced hypoglycemia. Looking at this a little bit further, then we looked forward and said, well, how does a weight-based insulin dose, if we were thinking about using such a multivariable model, how does this compare with a weight-based insulin? So here we plotted, this is a weight-based insulin dose, which is what we would use, which is part of the Flinders or the SALHN protocol of 0.5 units per kilo. On the x-axis is a 24-hour insulin dose, and on the y-axis is the difference between how much the patient actually required and the weight-based calculation. So to walk you through this, take this participant or this patient in the orange. So their actual weight was 98 kilos. The actual dose that they required was 37 units. But when we're calculating a weight-based dose, you would almost overdo them a little bit by 12 units because you'd calculate 49 units. And then in comparison, someone down here, so their actual weight was 85 kilos, but they actually required 183 units due to a range of other factors. But their weight-based dose was only 43 units based on a weight-only protocol. So you're underdoing them by upwards of 100 units and certainly putting them at significant risk of hypoglycemia based on what we currently do. If you look at the multiple variable-based insulin model, though, you'll see both that the range in terms of the difference is much narrower and that the slope of that line is much flatter going towards zero, which is kind of what we're trying to do to try to minimise the difference between what we're actually dosing to what the patient actually needs. And then taking the same two participants, so this patient then with their actual weight, with their weight of 98 kilos, using the multiple variable dosing, the variable dose is 38 units, so the difference is only 1 unit, and that other participant down here in yellow, so their weight of 85 kilos taking into all the other variables, so they would require 125 units based on a multiple variable based insulin dose, so the difference in insulin requirements has been narrowed. And just putting them side by side, the main difference, the main things to see is that one, that the slope is much flatter, and also that the range of differences is much narrower. So in summary, in hospitalised patients with hypoglycemia who are prescribed PRED for acute inflammatory illness, what we found was that a model comprising of weight, HbA1c, sex, diabetes status and diabetes medication will explain a significantly larger amount of the variability in their daily insulin dose. And incorporating these characteristics into calculations will hopefully improve glycemic control in this hospitalised patient group. And in terms of future directions as to where we'd like to take this, certainly want to look at kind of the prandial and the basal component, looking at potentially a randomised controlled trial that tests this model against the current standard of care, and then hopefully in the future even thinking about developing some form of online calculator to be able to access this faster. Definitely would like to thank all of the patients and the nursing staff and the physicians just on Flinders on level 6, without whom this study definitely wouldn't have been able to proceed. I'm very happy to take any questions.
Thank you. In the interest of time, we might let Angela off the hook for in-person questions, but feel free to ask her a question in the app or any other more old-fashioned way, maybe by email or any other contact. And we will then allow everyone to get to the break, which includes poster presentations. Thank you to all the speakers and for the great audience participation. Enjoy the rest of the afternoon.
I just want to welcome everyone here in the room that we have with us, those of you online and those that are going to watch this session later. It brings me great pleasure to introduce Audrey Taylor, who is a long-term staff member here at SALHN. She's an Aboriginal Hospital Liaison Officer within Karpa Narratendi, who's also training to become an Aboriginal health practitioner. And Audrey is going to provide our greeting to country today. Thanks, Audrey.
Nina Mani, which is hello, how are you in Kaurna language. We acknowledge the Kaurna people are the traditional custodians of the Adelaide Plains and pay respects to elders past, present and future. We recognise and respect their cultural heritage, beliefs and relationship with the land. We acknowledge that they are of continuing importance to the Kaurna people living today. We also pay respects to the cultural authority of the Aboriginal and Torres Strait Islander people visiting and attending from other areas of South Australia and Australia. Thank you.
To know me is to breathe with me. To breathe with me is to listen deeply. To listen deeply is to connect. It's a sound, the sound of deep calling to deep. Dadidi, the deep inner spring inside us. We call on it and it calls on us. We are river people. We cannot hurry the river. We need to move with the current and understand its ways. We wait for the rain to fill our rivers and water our thirsty earth. We watch our bush foods and wait for them to open before we gather them. We wait for our young people as they grow. The time for rebirth is now. If our culture is alive and strong and respected, it will grow. It will not die, and our spirit will not die. I believe that the spirit of Dadidi that we have to offer will blossom and grow. Not just within ourselves, but in our whole nation.
So we welcome you to today, and we ask you, we hope you come. We want you to be grounded. We want you to connect with each other and we want you to most importantly come with an open mind today. My name's Jessica Stott, and I'm the Research Coordinator for Aboriginal Health, and it is such a privilege for me to be facilitating this session today with a fabulous panel that we have here. We've got a great session planned, and we hope that this will be an interactive session. I'd like to also just provide my own personal acknowledgement. So I acknowledge this land of the Adelaide region is the traditional lands owned by the Kaurna people, and acknowledge our gratitude that we share this land, our sorrow for the costs of the sharing, and hope that we can move to a place of justice, reconciliation and partnership together. I pay my respects to elders past, present and emerging. I also just wanted to start, I've been doing a lot of reading in the research, and I came across a quote by an Aboriginal elder recently that said, "You've got 2 ears and 1 mouth. Why? It means you're supposed to listen twice as much as you talk." So in saying that, I want to introduce you to our esteemed panel briefly and let them introduce themselves. We're using all modalities today. Associate Professor Tamara McKean has been unfortunately unable to be with us, but she's been gracious enough to provide some pre-planned recordings, and we're also doing some virtual sessions with another panel member. So I'm going to start with Tamara, who's going to introduce herself.
Hello everyone. I'd like to begin by acknowledging that we are meeting today on the lands of the Kaurna peoples, the traditional custodians of the Adelaide Plains area. I'd also like to acknowledge the traditional custodians on the lands from which other people may be calling in from, and I'd like to pay my respects to elders, past, present and emerging, and pay my respects to other First Nations people who are joining us today. My name's Tamara McKean. I have a couple of roles here in Southern Adelaide that are relevant for the panel discussion today. I'm a Wallian woman, so I am not working on my country, and it is indeed a privilege and an honour to have been working on Kaurna country for the last few years. I'm a medical practitioner and I've trained in public health medicine, and so I have a number of hats that I wear. I'm currently the Associate Professor for Aboriginal and Torres Strait Islander Public Health at the Flinders University, and I do sit on the board of the Southern Adelaide Local Health Network. I hope that the video that we've shown to start the session has enabled all of you to feel grounded, to be present today, to come to the session with an open mind and with a caring intent. And I look forward to seeing how the panellists respond to some of the questions put to them by the audience.
I'd like to ask Dr. Kim O'Donnell, and just also acknowledge how grateful we are that Kim has stepped in at such late notice to be a representative on our panel. So Kim.
Thank you. Oh, goodness me. I'd also like to acknowledge this beautiful country that I've been living and working on for the last 21 years. I'd also acknowledge everyone here that's come, and our elders up the front here from other parts of Australia. And thank you very much, Audrey, for the beautiful welcome. Let me see, where do I start? Okay, so I'll give you a little bit of background about myself, and then I'll talk to what it means to me in terms of Aboriginal knowing, being and doing. So I'm a Mullingarpa Barkindji woman, or Noongar, from far western New South Wales. I'm a Barker woman, so my role is to take care of the Barker River, which is the Darling River. I grew up in Wilcannia. I have family still in Wilcannia across the country. I'm a mother, I have a son, and I have the most amazing blended family. My husband's Hungarian. We travel to Hungary regularly to meet with his family. I grew up and did most of my schooling in Dubbo. Also became a school teacher in about 1986. My first teaching position was Pepper Manati in Northern Territory, which is not far from the Daly River. And I spent 3 years there working with the Ngangikurungu people, and I still have that connection with the families there today. I went to Japan and taught English in Japan in 1989. As you can see, I've got a bit of an adventurous spirit. I get that from my family and my dad. I did that. I came back in the early 90s, lived in Sydney, studied Japanese at Sydney University, taught in Redfern. I also started a small business called Namaku Bush Creations, and Namaku in my Barkindji language means Mother Earth. I started that business in the early 90s. And then I became a flight attendant. I moved back to Darwin in 1994, based in Darwin, flying around Australia, did refugee charter flights for 7 years, and then I moved here to Adelaide in 2000 and decided to go back to university to do my Masters in Primary Health Care and a Doctorate of Public Health. My doctorate looked at how the government departments and government fund Aboriginal community-controlled health organisations across the country. And I was very honoured to work with an Aboriginal health organisation here in South Australia. So currently, I'm a public health researcher. My focus and my passion is in Aboriginal health. It's in ensuring that our families have equitable access to culturally safe care. And what does Aboriginal knowing, being and doing mean to me? It's my essence, it's my spirit, it's who I am, it's how I grew up. It's I also have a dash of Irish and German, but I grew up strong in my culture. I'm also the custodian of the Murruwinji lands in far western New South Wales, which was the first national park in 1998 to be returned to the custodians. I still travel to and from country on a regular basis, and I still have deep connections to country. And I guess for me, that knowing, being, and doing is the very essence of me. So I think I'll pass you back to Jess on that note. I think that was 3 minutes.
Thanks, Kim. Simon Windsor in the room, do you want to just give us a bit of your reflection too on, for those that don't know you, but also on the session title, what does Aboriginal ways of being, doing and knowing mean to you?
Yes, absolutely. Thank you, Jess. And I also acknowledge the Kaurna people. We meet on their land today. And my background as a medical scientist, when I graduated here at Flinders University, I moved over to the UK, worked there for a couple of years, and I'll tell you, Australians are they're a highly valued commodity in the UK, and I think part of that is to do with what it means to be Australian. I think we're a very resourceful country, and that is valued overseas. Then I moved back to Australia, and I had the opportunity to work in neonatal intensive care units across Australia and also in New Zealand. And that gave me an opportunity to work with Maori groups as part of Waikato and Wellington Hospital. And so that was my sort of first foray into working with First Nations people, and we modified our consent forms to include Pacific Island language into our consent processes. And I've been here at SALHN for the last 4 years. Now I'm manager of the Office for Research. And in more recent times, I've begun working very closely with Kapana Tindi and really listening and learning about their approach to care and what they're doing for the community here. So that's me.
Thanks, Simon. Annabelle, if you don't mind introducing yourself and what your reflections are on what this session title means to you.
Great, thanks Jess. I'd also like to acknowledge the Kaurna people as the traditional owners of the lands that we're meeting on, and pay my respects. So I'm Annabelle Wilson. I'm a non-Aboriginal person. I trained at Flinders Uni as a dietitian, and after working as a dietitian for a little while out in rural and remote communities in SA, I decided to do a PhD based on some of the challenges I experienced working as a dietitian in non-Aboriginal health, which were mainly associated with the differences in expectations between what I was expected to do by the health service I was working for compared to what the community were after. So my PhD looked at how non-Aboriginal health professionals work in Aboriginal health and what some of the barriers and facilitators are to working well with community. I now work at Flinders in the Aboriginal and Torres Strait Islander Public Health team as a senior research fellow, and my research has continued along a similar line. For example, looking at how non-Aboriginal and Aboriginal peoples can work together, how we can use strengths-based approaches in our research and practice to ensure that we don't perpetuate deficit approaches. So for me, Aboriginal ways of knowing, being and doing are a reminder to challenge the system which privileges Western ways of knowing, being and doing. They remind me to talk to other researchers and also the students that I teach that there is more than one way to know, to be and to do. They also remind me of the importance of acknowledging standpoint. So stating who we are, what we stand for, and what attitudes, perceptions, biases and approaches inform the way that we see the world. Because without acknowledging standpoint, it can be very difficult to see beyond what is dominant. Aboriginal ways of knowing, being and doing also remind me to explore Aboriginal knowledge systems, including traditional knowledges, how they have survived and continue to survive regardless of past and ongoing colonisation. And finally, they encourage me to allow time and space for things to happen and acknowledge that relationships are fundamental in this area of work in every way.
Thanks, Annabelle. I'm going to ask Dr. Simon Quilty, who is a specialist physician who's joining us from all the way from Alice Springs today. And yeah, I'd like to ask you, Simon, if you wouldn't mind just giving a brief introduction to yourself and your background, and then also a bit of a reflection on what Aboriginal ways of knowing, being and doing mean to you.
Thanks, Jess. I'm sorry that my head is so much bigger than the other three presenters and in no way reflects anything but a computer screen. So my name's Simon Quilty. I'm a physician. I've spent most of the last 20 years in the Northern Territory, but I'm born and bred in Western Sydney, sixth generation Australian with ancestors from England, Ireland and Jewish. I was very lucky as a child to spend a lot of time in remote parts of Australia, particularly the Northern Territory, and I think that's where my deep inquisitiveness into other ways of thinking began, and indeed that's why I did medicine. So I went to Sydney University, the first of my family lineage to go to university, and did engineering because I wanted to make my car go quicker to fit into Western Sydney, and realised that I wasn't really a petrol head, and I wanted to live in the Northern Territory and to make amends for the colonial process, which I was already very aware of from my childhood experiences. And so I studied medicine and moved straight to the Northern Territory, and I've been here pretty much ever since. I was the first specialist to work outside of Darwin or Alice Springs and set up a service in Katherine Hospital, and spent 8 years in Katherine working with Jawoyn, Dalabon, Yolngu, Waterman, Gurindji, Naranman, Naliwuru, Walpuri, Mudpara people, just to name a few of the tribes that I got to know reasonably well. And my entire experience of working particularly as an adult physician with adults and particularly older Indigenous people, has been one myself of a deep experiential learning process where my eyes have been opened to the ways of Australia's past, to the ways that people related to their land, to the way that ancestral lands shaped people and cultures, to the magic that you can often feel in an Australian landscape but are far from able to understand as a non-Indigenous person. I'll recount a little story for you. So I've just taken up a role as the medical advisor for Purple House, which is an organisation, a truly extraordinary organisation here that is absolutely community controlled. And I now have a patient who is an old Yankunytjatjara man from deep in the southwestern desert, southwest of Docker River, and he told me a story of when he was a small child. He's now in his 70s on dialysis, but as a small child he saw a mumu coming through the desert. A mumu is an evil spirit, and it was pushing aside the trees and the plants and doing ecological devastation. And he saw it coming from the distance, and it was a bulldozer, and it was Len Bedell's bulldozer. Len Bedell is celebrated in Central Australia for his colonisation as he bulldozed roads into remote places. And so this old man has told me stories about the first time that he met a white person, and it was a white person driving a bulldozer. And in 1981, when I was 7 years old, I drove down one of Len Bedell's roads. My father pointed out the brilliance of Len Bedell, but never pointed out to me the absolute devastation that it did to the ecology and to the culture of the Yankunytjatjara people, for instance. And so I'm incredibly fortunate for having had the opportunity to work with these incredible people who have been so deeply generous in sharing with me what I can only describe as a parallel universe. At first, it was like a tiny little pinhole that I could just see through, and I realised that there was something extraordinarily deep on the other side. I think that a lot of non-Indigenous Australians get that sense. But for me, in my role as a doctor working with elders every single day for the last 15 years, that pinhole's opened up to a small hole that I can really peer into and can start to truly fathom the depth and extent of it. And of course, I now fully appreciate that Indigenous elders had an ancient way of experiencing well-being that our society too sadly and too often completely fails to see. So I really greatly appreciate the opportunity to attend. I'm on Aranda land today and pay respects to those people that have never ceded this land. And thanks for having me.
Thank you. I'm going to pass it over to Tamara now to share her reflection.
So, what do Aboriginal ways of knowing, being and doing mean to me? Firstly, our ways of knowing are very much our songlines. Our songlines spread across this continent, and they are a way of holding our knowledge within country and holding our knowledge between people and country through geography and through an oral history that is both sung and spoken. This is a huge knowledge system, and it includes health knowledge. It's a health knowledge system that has existed for over 80,000 years in this place, and it has much value and is relevant to the health system of today. So our Aboriginal ways of knowing in health and healing are not actually something of the past. They are something for the present, and I believe they have something to offer our future. In terms of Aboriginal ways of being, to me, this is our relations with one another, relationality and connectivity. This is about living learning. Our lived experiences are part of our knowledge systems, and that's not just for one individual lifetime, that is over generations. It's country as our family, not just as a place that we might step upon. Our bonds with country are deep, and they mean that we feel what's happening to our country. Aboriginal ways of doing are very much about holding space. For a number of years prior to European colonisation, holding space was about holding that knowledge and holding songlines on country to be able to transmit that knowledge generation to generation. Now we are holding space and caring for country in a different way, not just in terms of people's health, but also in terms of climate and our environmental sustainability. If we have no environment, and if our climate is destroyed, human health will not exist. These things are intertwined, and Aboriginal people have known that for a long time. And so when we are doing our healing, we are doing it in an ecological sense. We are working towards holding space that heals ourselves, heals others, heals our nation, and keeps our environment healthy and sustainable into the future for our children's children.
Okay, great reflections there. I wanted to now turn the dial a little bit and touch on a term, decolonising, because I think it might be a term that might not be familiar to many of you. But conducting research with a decolonising lens is said to be critical to truly improving the health outcomes of First Nations peoples. This is something that Tamara is really passionate about, so she's going to talk to us a little bit more about this term and share how this approach can really reform the way we think about the design, development and delivery of health services.
Decolonisation refers to processes that centre First Nations worldviews and explanatory models, and in our case that means specifically explanatory models about health and healing for First Nations people. Decolonisation recognises that colonial processes, both previously and currently, has created structural, social and psychological impacts on the well-being of First Nations people. So decolonising health services is about taking account of all of those determinants of health, the multiple determinants of Indigenous health. And it also means that the service needs to engage with First Nations people about decisions regarding their health and the way that they want services to respond to their needs. This also means that a health service like SALHN actually needs to take action and to change the delivery of services and to use different models of care and to build up a culturally responsive workforce. And in doing so, to challenge unhelpful constructs that sit in the system as well as in people's minds about the way forward for First Nations health, both here in Australia as well as globally. It means engaging with the structural issues as well as being able to undertake healthcare interactions that enable non-assumptive diagnostic, reasoning and management approaches.
Okay, Simon, to you now in Alice. You touched on a little bit of healing within your first reflection. I want you to sort of talk to us a bit now about sort of the differences in and perspectives on healing within Aboriginal and Torres Strait Islander culture and our sort of Western way of thinking.
I'll give you an example, Jess, that I think describes it well. So from my perspective, I see Nunkaries practicing in Alice Springs Hospital there, and I know a few Nunkaries, and a few people have taught me some things that I'm allowed to know about Nunkaries and how that knowledge is attained, and it was all ecological. Indigenous culture is all based around the ecology that they understand so incredibly well. And Alice Springs Hospital is a really great place. They do fantastic work, particularly in the ALO and linguistics space. It's a very safe hospital. I dare say probably the safest in the Northern Territory, if not Australia. And because of the significant number of Indigenous patients in our hospital, seeing a Nunkery practice is common for most staff, but we often feel a bit perplexed about what is actually going on. So recently I was asked to MC an event to do with NAIDOC Week, where we had 3 Nunkeries, Aranda Nunkeries, come to the hospital to an audience in the evening to watch a movie about a Nunkery in the APY lands, and then for a Q&A session, which I nervously had to MC. And it was a really great session. There were about 100 staff at Alice Springs Hospital attended, and towards the end, the senior lady said, "You know, the problem that Western health has that we Nunkeries see all the time is that you guys heal the body. We heal the spirit primarily. That is most important to us. But we see that the way that you practice has nothing to do with the spirit." And I certainly know in my own reflections of my profession that it is an aspiritual dimension, but nothing can be aspiritual. I'm an atheist, but I'm not aspiritual. Particularly when you live in such an extraordinary environment and you can sense the timelessness of the land. And so when I reflect on my own profession, it is very mechanistic. It is very corporatized. It is like when you go and work in a hospital that you are part of a machine that can be replaced by another piece. For Nunkery healers, they don't suffer the same existential disillusionment that I see a lot of my colleagues suffer from in terms of the professions that we embarked on. And I feel that there's an awful lot that we can learn from Nunkeries and from Indigenous healing practices, for instance, bush medicines. And bush medicines work. I've seen them work in elderly people. I've seen how much they're valued by the community. And they are all about person, place and spirit. And it's really hard for Western people to know how to embrace spiritualism and wellness. But without spiritualism, you can't have wellness, I don't think. And I feel that we have an awful lot to learn from Nunkeries and healers and First Nations people around Australia and indeed around the world who are very synchronistic with the ecosystems that they know so well.
Yeah, some great thoughts there, Simon. I'm just going to stay with you for a minute because I guess for a lot of us here, you've obviously got the experience of and the privilege of working in the Territory where, you know, you've got a lot of contact and involvement with First Nations peoples. I guess for those of us here, perhaps that work in the medical field as clinicians that don't have perhaps the same level of experience that you do working in this area, are there any sort of key messages that, you know, or suggestions that you have as to how people might be able to begin that journey of embracing perhaps a different way of looking at healing and the way that we could deliver medical care differently?
I think the first and foremost thing is to understand who you are and what you take for granted about yourself. I say this to my junior staff all of the time. for me and for most people in the audience, we have most or many of our ancestors coming from a Socratic lineage, meaning that at some stage in the last 2 or 3 or 4,000 years, we've been influenced by Greek philosophy, by Western ideology that has become deeply ingrained in us. We live in rectangular structures with solid roofs that appear infallible to the future. We have become non-ecological beings. But there's no such thing as life on this planet that's non-ecological. And I feel that climate change and environmental disasters are beginning to tap us on the shoulder and remind us that we can't hide from the truth of our own ecology. And in that regard, before you can even start to reflect on how rich Indigenous ways of knowing can actually be, and they are extraordinarily deep. I believe that, from my experience, Indigenous societies have the most complex social structures of any social community that I've visited anywhere in the world, way more so than my own. To begin to fathom how deep it is, you first need to acknowledge how much of yourself is a social construct built around Western ideas. And so just even realising that perhaps a lot of the things that you take about yourself and your interactions with those around you, whether it being in the family space or in the professional space as a doctor or in any other social space, just starting to question that and realising when you see Indigenous people that hold on to, that have the luck of surviving colonisation and holding on to a lot of their culture, to understand how they view things that we view in an incredibly different way explains so much of behaviours that are often framed by Western healthcare providers and many other parts of Australian society as negative things, when in fact that they come from really unique and very real kind of social constructs of their own. So I think working with Aboriginal people, whenever you get the opportunity to, I personally, as a non-Indigenous Australian, I find it really enriching just to firstly start to realise how colonisation has impacted that person in front of me, and to learn from these people what their worldview is, and it certainly is a great place to reflect on your own perspectives, which is a good place to start from, I think. That's a roundabout way of answering the question. I'm sorry if it got a bit tangled up.
It's a big question, complex question. I wonder if anyone from the panel wanted to share their thoughts on that similar sort of theme. Annabelle or Kim, or around Western ways of thinking about health versus more Aboriginal and Torres Strait Islanders' approach to health and wellbeing.
I'm happy to talk to that.
Thanks, Kim.
I think that the original definition of health from a holistic environmental perspective was drafted in 1989 as part of the Aboriginal and Torres Strait Islander health strategy. And I think what we fail to do as human beings is, particularly in the hospital settings, is to just see health as being from a biomedical perspective, when really the biomedical perspective of health, where you see a person in body parts, in sections, and you might attend to one health problem and not necessarily relate it to another. I think what we need to do is to think of the definition of health and have a common definition of health that encapsulates first and foremost the environment, our life source, but also the healing, the cyclical nature of human life. And I would suggest looking at the 1989 definition. It starts off as Aboriginal health is, but if you took away Aboriginal and had health is, and if I could see that in that definition of health, when I walk into a hospital When I walk into the front of Flinders Hospital, or I walk into Alice Springs Hospital, I walk into the Royal Adelaide Hospital, if I saw that definition of health that's all-encompassing, then I think that could possibly be a way forward to help us all to understand that that's health. It's huge, and biomedical version of health is a part of that. So I think something like that would keep us all on track as humans, as the family here of humans. That's basically what I think of health. That's Annabelle?
Yeah, I'll add to that, Kim, in the sense that I was going to say that I think often there's a perception that, you know, that there's no crossover between different models of health. And I would say that, you know, different Aboriginal views of health, the biomedical model, etc., other examples don't have to be mutually exclusive. So, for example, if we take a knowledge interface approach to health or to research or to whatever, that allows us to picture different models crossing over, like circles crossing over. And in those spaces where those different models cross over, we have new approaches and new knowledge that wouldn't be created should we not look at things from those multiple perspectives. So I think it's about acknowledging that there are lots of different perspectives that can contribute.
Yeah, Simon.
Yeah, from my perspective, I guess the sort of recent observation is the way that sort of COVID vaccination's been rolled out, and some of the way that there's maybe been limited engagement in different communities, and that how that has impacted on the uptake. And so, if there's sort of more holistic approach, maybe that would have been more successful. And I guess from a sort of SALHN perspective, looking at some of the design features of the sort of some of the new spaces we have. So the wrap building has certain elements. I know that like arts and health, we've got arts and health here that's sort of 22 years old or 26 years old, and so I guess we're starting to see some of these green shoots. But I think maybe more emphasis on that would be the way to move ahead.
I think it might be a good opportunity, Simon, from Alice. You spoke a little bit before about the ways in which you think Alice is perhaps the most welcoming hospital that there is, at least from your perspective and experience in hospitals. And I wonder, what is it about Alice Springs that you think makes it so welcoming? And is there some things in there that we can sort of learn from as an organisation?
Yeah, so I've worked in every hospital in the Northern Territory. I've worked in quite a few in New South Wales as well, but most of my time has been in the NT. And Alice Springs really shines the light on how to provide safe Western care. I think that it has a lot of work to go in terms of addressing the issue of biomedicalisation, and I feel that a lot of this vaccine hesitancy, as the panel's talking about, is not just amongst Indigenous people, it's amongst everyone. And I think there's a great community-wide acceptance that there's something missing from healthcare. So I don't mean to address those issues, but at Alice Springs, what this hospital does really well, and this was well before my time, and I'm not quite sure why it happened, but senior levels of the hospital realised that they had to engage with people from a very different culture, and then over time the hospital has populated with a very progressive bunch of people that deeply recognise the need to engage in culture. So just the fact that I had over 100 people turn up to a conversation with the 3 nunneries was very impressive. Mm-hmm. The staff that come to Alice Springs self-select, they know what they're in for, and they come with an open heart and open minds. So our Aboriginal liaison officers are the heart of the hospital, They're at the very front door of the hospital. There's 10 of them. They speak every language. They are a team. They move around as Indigenous people, every single one of them living in a town camp house with their community. They know their community intimately. They accept the Western biomedical, as does everyone, because it does prolong life. They have reservations, which they'll openly express, but they are a team together and they walk through that hospital proudly. And everybody in the hospital, right to the general manager and the executive director of medical services, acknowledges that without them, we are lost. We could not provide the care that we do. What is really exciting, having worked in other hospitals in the Northern Territory and seen many patients very afraid of coming into hospital because of their experiences and lack of support, is that we have patients that come into Alice Springs Hospital because they want some respite from their very hard lives, which I think is a wonderful indictment that we're doing quite a lot of it well, and not perfectly. There's a zero tolerance of racism or even policies that kind of alienate Indigenous people. It's not perfect, but it's very good. And there's a striving by everybody and a recognition that we're here to serve an Indigenous population and we want to do it as best as we can. So I think that's where it comes from, but it doesn't quite address the broader issue of where Western biomedical health is failing to make people well.
Mm.
In order to implement different models of care, we need to think about the role that Aboriginal and Torres Strait Islander health workers and health practitioners and liaison officers play in multidisciplinary team approaches to care. We're very fortunate at SALHN to have had Kapa Narantendi, which in Kaurna language means to support, to heal, since 1997, and that unit has offered an invaluable service to many Aboriginal and Torres Strait Islander patients, particularly at the Flinders Medical Centre, but not exclusively too. The role of Aboriginal and Torres Strait Islander health practitioners, health workers and liaison officers in hospitals is different to the way it is in primary health care. They are often involved in being able to ensure good communication occurs, particularly around issues of informed consent for undertaking different procedures, and they are also now part of the Australian Health Practitioner Regulation Agency. We have an opportunity at SALHN to really grow this workforce. We've had the Lighthouse project with the cardiac unit and the use of Aboriginal and Torres Strait Islander health practitioners in that unit to enable continuity of care, to ensure good medication management, to facilitate timely access to operations as well as proper discharge. And the role that those practitioners play has actually been given a SALHN award in the past. So we have good examples to work with and good things to build on. We do need to make sure that the rest of the workforce properly understands the role of Aboriginal and Torres Strait Islander health practitioners. They are not social workers. They have a different role that is about a cultural exchange and ensuring that both the caregiver as well as the care receiver are able to engage in an interaction where needs are understood the different priorities of both the patient and the clinician can be shared, and an agreed approach and a way forward that is cognizant of the different priorities can actually be reached. We need Aboriginal and Torres Strait Islander health practitioners within our hospital system if we are to deliver on patient-centred care, which is a core component of quality health care.
Okay, Annabelle, I'm just going to ask you now. Now, you mentioned, the importance of acknowledging standpoint. Can you sort of elaborate for us on this, particularly as a non-Indigenous researcher working in this space? What does this truly look like, not only in your work life, but also your day-to-day life?
Sure, thanks, Jess. So for me, standpoint is about acknowledging who you are, what you stand for, what influences how you see the world. So that can be both, that is both personal and professional. So as an example, for myself, I am a non-Aboriginal person, I'm a mother, I was trained primarily in the biomedical model. I've learnt to see things differently. I work together with Aboriginal and Torres Strait Islander people to strive for outcomes. I see things, I like to try and see things in a strengths-based way rather than a deficit approach. So these are just examples of things that influence the way that we see the world. And as a non-Aboriginal person working in Aboriginal health, it's vital that I declare what my standpoint is. And often I will have other non-Aboriginal people speak to me about being fearful around working in Aboriginal health. And certainly a lot of my research has shown that can be the case. And what I always say is that, it's not about whether or not you're the right person to do the work or work in the space. It's about making sure that you acknowledge your standpoint, because I think that if you acknowledge your standpoint, then you're showing the starting point of which you're coming from, and that's what is really important, to be able to move forward and find shared ways of working.
Yeah. You're nodding, Simon. Did you have anything to add to that? Or?
Yeah, I think they're really wise words, Annabelle. I think acknowledging who you are and what you stand for. I think health can be full of deeply divisive politics and complexities, particularly Indigenous health. But when you know who you are and why you're there, and you express that really clearly so that everybody can understand how you fit into the puzzle of it all, then it really aids to collaborate and to collectively get around working with what needs to be done.
Yeah, for sure, Kim. Yeah, please.
I also think that's the first step in terms of creating a relationship, creating a friendship. I think what's always happened in the past is there's this huge expectation for Aboriginal people to constantly give of themselves, and I think it's really the very first step to create friendships and partnerships. But it's also truthful. It's about speaking truth to your culture, to who you are, and it goes both ways. I think also when we talk about our standpoint and the impact that our cultures may have on each other, it also makes it easier to trust someone. This is who I am. this is why I'm here. I'm a mother, I'm an auntie, I'm interested in this topic, I'm interested in that. And you start to connect as human beings rather than having the barriers up of distrust. So, and that takes time. It takes a lot of time to, I think, develop trust given our history, which has been a very divisive, very colonising history that has had a lot of, and created a lot of damage and separation for our families. So I think it's really important for health professionals working with Aboriginal people to actually step up and or step aside to enable the Aboriginal leaders to come through. And sometimes that way of working is a flat structure. I think the top-down approach for working with Aboriginal people, it doesn't work. It's a very colonial approach, and I think if we think of when we're working in that knowledge interface in the space where it can get tense and conversations can get quite aggressive sometimes, but that's why it's very important, I think, to say straight up, this is who I am, this is where I've come from, this is why I'm here, and this is why I really want to work in this space.
Yeah, that's good. So given it's Research Week, we might just touch on a little bit of some of the research that's been sort of done in this space. We know that research has a pretty negative connotation in the way that it's some approaches that have been taken with Aboriginal and Torres Strait Islander people, that approach of research being done to people. And I know at the consumer session that was spoken quite a bit from the consumer lens as well. So I'm interested to sort of put to the panel, though I think there is a shift and we are learning, what approaches aren't, successful and not the approaches not to take. And so I'd be really keen for the panel to sort of share some examples perhaps of some research that they've either been a part of or that they've witnessed that have been successful, and to sort of just describe a little bit about what was it about that approach that research, made that successful. Simon, do you mind if I throw it to you to kick that off?
Yeah, so I have been involved in research for 20 years now in various ways, and I use it as leverage, but I guess it has to come from the community and it can't be done on the community. There's no need to measure parameters of biological demise that are perfectly expected in the social circumstances. So, for instance, I'll give you one example of something that I feel deeply vexed by, and that's the millions, tens of millions of dollars that are currently being spent on the development of a vaccine for rheumatic heart disease. So this will mean that children get more needles through the trial, there'll be more people being enrolled, they'll all be Indigenous, it'll all be for a vaccine against rheumatic heart disease. My grandfather died of rheumatic heart disease when he was 41 years of age, and he was a white fella, and he grew up in Depression era Sydney in a house with 10 other kids in it, and he got rheumatic heart disease, and we know it runs very strongly in families. None of his children or grandchildren or great-grandchildren or great-great-grandchildren acquired rheumatic heart disease, and it's because the social conditions, which were the fertilisation of his pathology, were resolved in 1940. And for all non-Indigenous, for the vast majority of non-Indigenous Australians, we don't live in grossly overcrowded houses like a lot of the people that I know here in Alice Springs and Katherine, 20 or 30 people to a 2-bedroom house. I kid you not, this is happening in Alice Springs in hundreds of houses tonight and tomorrow night and onwards, and it's not getting any better. And so to develop a vaccine is kind of like saying, don't worry, we'll give you a vaccine, we'll give you a needle, so you don't get the virus, that you don't get the heart disease anymore that has a 40% chance of killing you before you're 40 years of age. We'll let you have your conditions of extreme overcrowding and we won't fix them because now you've got your vaccine. And so I think if you asked Indigenous people in places like Alice Springs or Katherine, where they'd want $100 million spent on the development of a rheumatic heart disease vaccine, or whether they'd like the housing addressed, the answer is clear to everybody. So you have to know your community, and it has to come from community, and it has to be joyful and exciting, and you have to bring Aboriginal brothers and sisters along with you in a way that's really fulfilling, and it just feels right. You know when you're onto it because it feels right, and the people that you're working with are excited about it, and that it feels like it's not your voice, it feels like it's a collective voice, and you don't feel like you're subjecting people to more Western biomedical kind of protocols.
Yeah, great. Good thoughts there, Simon. Kim, you've done lots of research, but I guess one that's quite relevant perhaps to a number of the audience here is around the work that you're currently doing within the kidney space and the wonderful work that you're doing there. And I wonder if you might just share a bit of that work with us.
Sure. So my colleague, Dr. Janet Kelly, was successful in getting a 12-month funded grant from Health Translation SA, and due to COVID it was extended. But basically I came on board with that team 2 months into it, and the aim of action, which is to improve access to transplants, improve culturally safe care for Aboriginal families and our patient experts from rural and remote areas, and also metropolitan in South Australia. So we centred the Action Reference Group in that project, and the Action Reference Group is made up of the most amazing, strong people I've ever met. The group is made up of patient experts. We don't call them customers, we don't call them clients. They're our patient experts, and the patient experts in this team, some have had transplants, some are awaiting transplants, some have had their second kidney transplant, some are on dialysis. We've just engaged and have a young mum now with her little baby who was born with end-stage kidney disease. And what we've done just recently is we won an ideas grant from NHMRC. So we have now 5 years of funding to improve access and equity and services, and to ensure that renal space and the journey from rural and remote places is the best that it can be for our families. So we were quite excited that we managed to get that grant. As you know, if you've applied for NH and MIC grants, it's a lot of hard work, and so this will be the first project, NH and MRC project, that I'll be leading with my amazing patient experts. So what we've done is we've totally flipped it. So we have our associate investigators on that grant. The associate investigators are the nephrologists, the kidney nurses, other Aboriginal colleagues across Flinders University and Adelaide Uni. Tamara McKean is an associate investigator on that project as well. So we flipped it, and we're centring patient experts, and we also have a team of researchers which is also made up of the nephrologists and doctors, and we have a group of stakeholders as well. So that 12 months funding, we spread it. We made sure we spread that, and we're very grateful that we were successful with the NHMRC grant, which will enable this work to continue for the next 5 years. And what we want to do is embed culturally safe practices within the renal units and across the some people call it the corridor from Northern Territory down to South Australia, We call it the spine of Australia, but we want to ensure that we improve services and access and equity, and that it's directed and led by Aboriginal patient experts and Aboriginal public health researchers. So we're very excited, and we've just got our team together, and I've got a young member here, Kanisha. Turn around, Kanisha, and give everyone a wave if you like. Kanisha is our admin person, and Kanisha, I shamed her up too.
You have shamed her, yeah.
Yeah, so Kanisha's joined the team and is also a graduate from Adelaide University. So Kanisha's here to give me strength.
I guess I can comment on, so I was recently out at Lyell McEwin Hospital, and there was a session there on social determinants of health outcomes, and the speaker was, I think it's Professor John Lynch from Adelaide Uni, around sort of child protection discussion and disadvantage and advantage sort of communities. And one of the key improvement elements from that work that he did, sort of assessing all of these different programs, were social investment programs. And so it's that sort of thing of actually sort of building capability within community to to sort of determine their own destiny as well. So I think that's...
I also think that NH and MRC, in terms of how they distribute the research funds, there's been a lot of change over the last 20 years. I came into this space as a student with Annabelle, and we've supported each other along the way. I think that there's been a lot of change in terms of NH and MRC valuing the knowledge and the experience that Aboriginal people bring, but it's also been a whole lot of hard work by Aboriginal people, and it's taken around 20 years. And I have to say, I'm still not happy with the way the research funds are distributed. It was a big fight from the beginning to ensure that a percentage of the NH and MRC funding actually goes to Aboriginal researchers. Initially, when that happened, it didn't. It went to non-Aboriginal researchers, mainly elderly white men. And so that I've seen change over the years, but there's still a lot of work to go, and I still think we still have a lot of work to go in terms of how the fundings are distributed. So, yeah, we're in it together.
Annabelle, sort of through a lot of the discussions that I've had, I get the sense that, and I think you touched on it earlier, that a lot of people, they understand a bit of the why here, why gaining more knowledge in this space, or, you know, doing things perhaps to help improve health outcomes is important, but there's that sort of fear, if you're non-Aboriginal to non-Indigenous, to kind of step into the space, or it should really be left to Aboriginal people because they are the true experts. So what would you kind of suggest or say to those people that sort of feel that, you know, uncomfortable, sort of not sure where to take it, or kind of, yeah. Do you have any sort of...
Yeah, well, I guess I'd start by saying that feeling of discomfort is normal, and I would encourage people to embrace it and explore why they feel uncomfortable rather than to just run away. We know that, you know, for some people who haven't done it before, acknowledging standpoint and really being forced to think about what drives who you are can be uncomfortable. It can also be uncomfortable to think about and really acknowledge colonisation, and that colonisation is not something that just happened once and is over, that it's something that continues to happen, and critically considering, you know, what, whether how you play a role in that can be uncomfortable. So in those cases, I would encourage people to use a process of reflexivity, which some people often use that interchangeably with reflection, but I think they're quite different. So reflection is about acknowledging the way we're feeling, whereas reflexivity is really about looking at what we're feeling and why we're feeling uncomfortable, unpacking that, and then thinking about what we're going to do differently based on what we've noticed. So constantly being reflexive is one way to move through that discomfort. I would also say that, yes, there is a space where Aboriginal researchers may work solely with other Aboriginal researchers. However, I think there is also a need for non-Aboriginal people to stand alongside Aboriginal people and legitimately work together and collaborate. And that often involves giving up power and saying, I want to work with you, I want to hear your perspectives, rather than just going in with your own and saying this is the way things need to be. So I think as long as a non-Aboriginal person is willing to do that, then there's huge opportunity for them to work in the space.
That's great. I'm just going to hand it over to you guys now, because I'm conscious of time. I feel like we could happily be here for another, couple of hours. But, are there people? I encourage those that are online to sort of interact with the Hoover app for any questions they have. Or those of you in the audience, we're happy to sort of take them from you now. Does anyone have anything they want to ask? Dom.
Thanks everyone for a great panel discussion. My question is, for non-Indigenous researchers, what is the most important thing to consider when conducting research with Indigenous peoples?
Kim, have you got something, or Annabelle?
Or we can co-answer it, or perhaps Simon.
You can all have it.
My answer was, why are you doing it? Make sure you know why you're doing it. And it can't be for your reasons. It's got to be for the community.
Yeah.
I'd also add to be honest, and not to get stuck in the mud. So don't be fearful of stepping forward. Don't be fearful of having a conversation and sharing your ideas. My advice, I think the other advice from me is we're all going to make mistakes in this space. We'll all make mistakes. You might get roused. I get roused all the time. It's not just a black fella, white fella thing. We're all going to make mistakes. But I think what is important is when you make a mistake, admit it. Be open. Be able to discuss why you made that mistake or why you made a decision based on your ideas. And I want to just share with you a mistake I made when I was presenting back in the early days here at Flinders University, and I was learning all about research, and we had elders come from Spencer Gulf, and one of the elders was a mentor of mine, and we had Aboriginal researchers come from Spencer Gulf to see Ernie Stringer talk about action research, and we had invited him to come and talk about action research because the big terms in research was really, doing our heads in. So Ernie came along and we co-presented with him, and the elders were sitting up the front. And I was really nervous, and there's a whole mob of people in the room, including the head of health sciences at the time. And I failed to acknowledge our elders and our researchers that had travelled from Ceduna and from Port Augusta and Port Lincoln, and I failed to acknowledge them first. And halfway through the conversations, I was like, oh, geez, what have I done? So I followed the auntie out the door, and I tapped her on the shoulder, and I said to her, Aunt, I'm so sorry. I should have acknowledged you and others that have come. You've travelled the longest distance to be here. I should have acknowledged you, and I didn't. And she turned around to me and she had that cheeky little grin on her face. And she looked at me with a twinkle in her eye and she said, "That's all right." She said, "It's only a mistake if you make it twice." And that advice I take with me in everything I do. And she could have roused me. She could have got angry, but she didn't. And that's what she said. And for me, that was like, well, okay, I've made a mistake, I've admitted it, but if you make a mistake, it's only a mistake if you make it twice because you didn't learn the first time. In other words, don't make it again. Yeah. So, those are the moments that I think, and that's the advice I have for you. Don't get stuck in the mud. There's going to be times when there's going to be tensions, there's going to be difficult conversations to be had, but at the end of the day, it's everyone's responsibility to ensure that space, that brave space where we're all going to feel vulnerable, it's everyone's responsibility in that brave space to ensure that everyone's safe and to be respectful and to take the time to hear from each other. And not to always expect the Aboriginal person in the room or the Aboriginal person to have the answers, because we've done our time. We've given so much of ourselves, and our elders have given so much of ourselves to actually find and have a voice in our own country. And I want to pick up on what Tamara mentioned yesterday, truth, treaty, voice. It is about changing the Australian Constitution. It is about thinking bigger. It is about the environment. It's about climate. It's about the planet. And I don't think we can work in our little, bubbles. We need to be open and honest in that space and keep working on ourselves.
Yeah. In the absence of talking to someone, to someone who is Aboriginal or Torres Strait Islander, are there some useful things, guides? I mean, you can't certainly, and I'm not suggesting you can learn it all from reading. You certainly do need to have those enriched conversations and interactions. But are there some tools that you would recommend, Annabelle? Are there some go-tos that people can, to get some further understanding as to some of this kind of thing, the right approach to take that you would suggest?
So I would say do your own research. Don't expect Aboriginal people to give you all the answers or give you all the information. There's lots of resources out there. There's lots of, popular media that people can use to learn. It's great when you do have a mentor, but I would say that it is non-Aboriginal people's responsibility to know about history, to know about colonisation, and to realise how that continues to have an effect.
Yeah. I've got a question here on the app. Extending that, what is the aspect most often done wrong?
Well, I can just mention something. So one of the things I was going to say to the question before is not letting deficit be the driver of your research. So I think that one thing that is often done, whether it's right or wrong, is open to debate, is that we're all aware of the discrepancy in health status between Aboriginal and Torres Strait Islander and non-Indigenous Australians. In the kind of research world where we have to justify, the problem, so to speak, and highlight the gap to get research funding, it can be very easy to buy into perpetuating those deficits and always reporting on where things are wrong, what the problems are and what needs to be fixed. So that is often done, and that represents a form of ongoing colonisation because it's constantly highlighting deficits without talking about strengths. So I think that researchers need to reframe the way in which they talk about those issues rather than just always highlighting deficits. And there are ways to do that.
Yeah.
I totally agree, Annabelle. And it reminds me of our patient experts, and that we centred in action, and words from Rani, and you see her coming through in the next 2, 3, 4, 5 years. She said, we're just not. We don't want to be seen just as patients or clients or customers. We don't want to be seen as just surviving. We want to thrive, not just survive. And in order for us to make that happen, we do have to change the way we research on Aboriginal people. Actually, we research with and alongside Wikaraway in Barkindji, with and alongside, but also understand that, and know when to step aside, when to step forward, when to step back, and work with Aboriginal people so that we're able to lead our own research, develop our own questions. And we certainly have a much bigger pool than 20 years ago in terms of quantitative and qualitative researchers across the country, from grassroots to government to high level to in between. So we've also got a certificate for research course being written to grow more Indigenous researchers across everything, whatever they want to research, just not in the health space, but in other places. There's a Lowitch Institute which started off and was funded initially by, and it started as the Cooperative Research Centre for Aboriginal and Torres Strait Islander Health. It's now grown into the Lowitch Institute, which has just become community control. If you go onto their website, you'll find a whole lot of amazing, rigorous research work that's been done and led by Aboriginal people, and also alongside Aboriginal people are our non-Aboriginal researchers that have walked the walk and talked the talk. And I think to expect Aboriginal people to come on board in a project or in a project that you've got funding for, and you've not involved them at the very beginning, or not have evaluation woven through that, that's not good as well. But I don't want you to run from this space. I guess it's really important. This is new. This is new ground here. It's probably not new for us because we've been singing out for a long time. To have people walk beside us and to value what we bring, but I want you to stick with it. And as Annabelle mentioned before, you will feel vulnerable, you will feel shamed up, you will, like I just done with Kanisha, bless, she's going to go from that. But you will, and it's really what we've been feeling since 1788. So come on board for the ride, and let's do it properly and let's do it respectfully.
To reiterate, follow on what Kim's saying around that change aspect. We're talking about sort of ecology, racism, and change, and I guess people are resistant to change sometimes, and I guess it's about the language as well, and maybe growth. Everyone wants to grow, and everyone wants to, so it's the mindset and how we approach these sort of major society issues, and to try to eliminate that resistance and, achieve great things together.
Thank you, Simon. Very conscious of time. Kim, thanks for sharing that last message. I think that was a really good take-home message for people, who might be, feeling that sense of fear, but that, that open acknowledgement that we're here to work together to do better. No, that's all right.
But it's going to be rewarding. And I just want to sorry, I've cut you off again, just sorry about that. That's all right. I just want to also say that, look, government makes the decision. They've got the budget. They make the decision about where funding should go. And I just want to remind you that the Aboriginal community-controlled health organisations, which were established over 40 years ago, and there's now around 140 of those across the country, they were initially established because Aboriginal people and Torres Strait Islander people were discriminated against and weren't provided the service in hospitals and in health centres, rural and remote, and in the cities. Now, these organisations have been established and operating for a long time, and when we talk about decolonisation, we talk about, people may not necessarily understand what that term means, but they are certainly doing it. And so this is doing it in the Aboriginal way that Aboriginal leaders know what needs to be done. But I think, government needs to fund Aboriginal health services, and not from a million little buckets of money and then expect them to have multiple, reporting requirements that take them away from being on the ground. It's to actually give them control and power to put that money where they see it needs to go, because one Aboriginal health service is not going to have trachoma problems as another health service. So give the power and the control back to those services and fund them and support them and walk alongside them.
Thanks, Kim. I'd made a personal promise that I was going to finish the session with a clip from Tamara, just to acknowledge her involvement in the session. But I do want to give it also Simon from Alice, who's been so gracious in joining us today. And I think we have, obviously as part of our organisation, we do see a lot of remote Aboriginal and Torres Strait Islander patients, and perhaps, patients that you're involved with, Simon, who you have in fact referred for, specialist treatment here. And I think that the relationship we have with our colleagues in places like Alice and some learnings that we have, I think it's a really important step in sort of taking this conversation further. But Simon, I'll hand over to you now. Oh, that is close. To just provide some concluding remarks from you, and then I'm going to flick to Tamara, and then.
Thank you, Kim. Look, I'm really glad that you said those words. I think that being unafraid of making mistakes, and from my own point of view, I have a few Indigenous older men who have very much tolerated me as I've stumbled through my journey, and they laugh about the skinny little white man from Sydney, and how he's turned into a proper man out in Alice Springs. And reflecting on my invitation here and my deep friendship with people like Tamara, working in Indigenous research in a really authentic way is incredibly enriching and rewarding. I have friends like Tamara who are not just friends, but role models, mentors, and stalwart supporters, because we're all in it together. And it is a place of great joy to be in, and I feel like I'm part of the family. And it's a wonderful place, but it's about being courageous and seeing through the hard times and keeping your integrity. And the fact that this has been arranged by Tamara speaks volumes to what your organisation is striving to achieve, and just striving is the first place to start, and it all comes naturally once you've got that Indigenous leadership showing the way.
Thank you, Simon. Okay, I'm just going to play the clip from Tamara now, and thank you all so much for coming. Really appreciate the support, and for those that are online. And I really hope that this is the start of many more conversations in this space. And yeah, it's a real pleasure and privilege to be here.
One of the most challenging aspects of hospital care for Aboriginal and Torres Strait Islander people is understanding the way that racism affects the health and well-being of patients in hospital through both institutional racism as well as implicit bias. It is very clear now that institutional racism and implicit bias kill Aboriginal and Torres Strait Islander people, both in the health system as well as in the justice system. There's a range of coronial reports that demonstrate the way in which implicit bias and institutional racism contributed to the untimely deaths of Aboriginal and Torres Strait Islander people. Specifically, the untimely deaths of Ms. Williams at the Tumut Hospital and the untimely death of Miss Dew in Port Hedland. I would encourage you to review those coronial reports and to understand why and how the coroner came to those conclusions. We really can't waste time, nor energy, nor our resources on debating whether racism exists or not. And we shouldn't be distracted from critical issues like Black Lives Matter by responding with statements like all lives matter. Of course, all lives matter. It's just that our lives, Black lives, are in real danger. And a just society, and a society that embraces equity, will acknowledge that and get on with the business of achieving social justice and reconciliation, and ask and answer questions about why and how things can be done better. We need to get on with the job